Very much. Mr. Dazi already mentioned that I'm going to talk about some of the microbial engineering work that we have been doing at International Center for Genetic Engineering and Biotechnology, especially to valorize agricultural residues to produce advanced biofuels. Just to give a brief background why here, especially in India, we are so much looking after the valorization of agricultural residues. We are being mainly agricultural country. We produce huge amount of biomass, and they are not really utilized efficiently. Many times they are burnt as well. So we want to make sure that we generate some useful product out of these waste that are being harvested and not utilized. And also we want to protect our environment by asking people not to burn it. And it's also important to understand that when we use fossil fuel as a fuel, then we generate huge amount of carbon dioxide in the environment. Worldwide, it is 35 billion metric ton of carbon dioxide that is being released, and they are mainly coming from the usage of fossil fuels. So there's a lot of effort from different countries all over the world to ensure that it is replaced with some. If you want to protect ourselves, the health of the people are very important, and therefore it's better to talk about precaution better than cure. So it's better to stop releasing the new carbon dioxide. And therefore, we believe that this particular molecule that we call biofuel, it's going to solve a lot of issues because mainly it is carbon neutral. They are being captured in our lifetime from the environment and then use it as a fuel molecule and convert it into the biofuel, which basically can be used in the engine. Now, at this moment, there are some biofuels already used in the, let me just change the pointer setting. Yeah, so there are biofuels that are present in the market. India already achieved 10% blending of the biofuels in, for example, in the form of bioethanol in the petrol. And these are mainly coming from the first generation. So, for example, if bioethanol are produced, they are done through the microbial fermentation of sugars, or if the biodiesel is produced, they are done through the transesterification of oil. And we want to avoid this because these first generation biofuels are not very sustainable because of the food versus fuel issue. So therefore we want to shift something beyond the first generation biofuel, and we call them second generation or third generation biofuel. So second generation is basically those part of plant which are not eatable. So that's the first criteria, and going away from first generation. So any agricultural residues or forestry residues that are left behind after biomass, because those agricultural residues, they are basically composed of lignin, hemicellulose, and cellulose, and therefore they are also called lignocellulosic biomass. We process them. There is a simple flow diagram here telling that how these residues can be converted into fuel. So basically, these hemicellulose and cellulose are polymer of sugar. So if you are able to remove lignin through thermochemical pretreatment, lignin is phenolic compound. Then these cellulose and hemicellulose can be converted, can be digested into sugar, simple sugar, glucose, xylose, etc. And these are done by enzymes. So cellulase, hemicellulase, and then these simple sugar can be fermented to ethanol. So challenge really is at multi steps basically, but the enzymes basically become a key criteria here because they are very expensive, and therefore overall the cost of enzyme becomes very high, cost of process become very high. And we also have a issue like C5, C6 fermentation because the sugar is not just glucose. There are xylose and other sugars which are not fermented by yeast, and therefore engineering yeast or bacterial species that can ferment all sorts of sugars are also important. And then lastly, ethanol can be made by fermenting sugars, but ethanol is not the best fuel as far as comparison with the fossil fuel are concerned because of its low energy density and hygroscopicity. So we also work on they become fiber because of the linkage of glucose, beta 1,4 linkage. The linkage allows the glucose to become linear in their chain, and the chain can sit over each other, making a very strong hydrogen bond. And this makes the overall process very, very, difficult to digest. And we can correlate with the polymer like starch, which is amorphous, they are branched, and while we eat, our salival enzymes, glucoamylase, amylase, can digest those starch into glucose, as against the cattle, who has to sit hours and hours to digest these molecules like cellulose, hemicellulose into monomeric sugar. And therefore, this crystallinity of cellulose becomes very, very important in terms of looking at enzymes. But of course, there are enzymes that can digest those cellulose, and we call them either exocellulose or cellobiohydrolase or endoglucanase or different second cellobiohydrolases. These generates the oligomers. There is a LPMO monooxygenase that generates a kind of nick in the middle, so that we have more and more oligomers and dimers generated. And eventually, beta glucosidase will convert these dimers and oligomers into glucose. Now, these we call them as a core enzymes, and these are not, I mean, these are it's not one or two enzymes like start getting digested by amylase and glucoamylase. There are like hundreds of enzymes which are basically important to hydrolyze these. And therefore, it's very important to find out a good platform that can produce those many enzymes that will be able to digest agricultural residue. Now, we start looking at many, many different kind of platforms. For example, a good platform at that time when we started looking at it were insects who were destroying the plant, for example, termite or cotton bollworm or rice stem borer. These are something very typical of the insect which destroys the plant, and the gut of these insects have microbes that help them to destroy the plant. So we looked at cellulases, hemicellulases, and many other enzymes, and we characterized them. But we realized that it's very difficult to get these bacteria produce large amount of enzymes. So eventually, fungi is microbes that can secrete large amount of enzymes. It's very difficult that these microbes secretes out enzymes eventually for the commercial purpose. So we looked at fungal isolates. We did a huge high throughput screening with the help of robotic liquid handling system, and we started characterizing enzymes that are coming out of it through the screening process. Now, we made a small metabolic model so that we can screen these enzymes based on whether they are producing any specific kind of enzymes, or they are composition of enzymes or concoction of enzymes, or they are actually able to hydrolyze biomass more efficiently. So we integrated all this in a small ethanol, second generation ethanol enzyme. And the mainly Novozyme whose enzymes are available with some of the partners. So we tested Novozyme enzymes along with some other enzymes that were available, and we found that this particular species is producing very good quality enzymes that we should be working on. So we started going deeper into it, because eventually, if we want to enhance the quality of enzymes and quantity of enzymes by the particular fungus that we have selected, we need to understand the biology and physiology of this particular fungi very, very importantly. So we looked at genomics and proteomics of the kind of enzymes that were produced by this fungi. We also found that this particular fungus is very, very unique in terms of producing enzymes. It senses what is there in the environment and produce enzymes accordingly. Then we characterized a couple of specific enzymes coming out of the important one, which was secreted by the fungus. And we also looked at specifically one of the substrate that are available in access, which is sugarcane bagasse. So eventually, the enzyme composition that gave us idea about what is it that making this strain so special. We realized that this exocellulase, which is GS7, is produced in a this particular enzyme, especially in terms of hydrolyzing the crystalline cellulose. So, for example, it can sit on the fiber chain, pull the chain up, and then walk on the fiber so that the chain goes towards its catalytic center, and then it cleaves the dimer of glucose, which is called silobiols. And when we compare the kinetic parameters with the well-known available trichoderma resi celobiohydrolase, we realize that the enzyme that we were working on had almost 5 to 6 fold higher catalytic efficiency and much less inhibition from its product, which is silobiols. So we looked at the model of the enzyme, and we could find out that the tunnel structure and the exit loops and all that are important for achieving these efficient biomass hydrolysis that it was doing. Then we started looking at engineering aspect, because we had this fungus in our hand and we wanted to improve the efficiency of the enzyme production. We wanted to see if we can transform this fungus, and we realized that it was very difficult because it was tolerant to most of the antibiotics that are used in the lab. And we did some transcriptome studies to realize that there are a lot of drug efflux pump that is produced by these, because of which they are tolerant. And we found a way to block these drugs. One in the fungus, we figured out the protein structure from there, and then we designed the cassette to delete this most of the portion of MIG-1, and we realized that our enzyme producing capability has increased by more than double. We also characterize another important enzymes which are produced by this particular fungus. It is LPMO, which is lytic polysaccharide monooxygenase. It is basically, it makes more end in the middle of the cellulose chain for cellobiohydrolase 1. So we co-express these two in the background of MIG-1 deleted strain, and we slowly improve the ability of the fungus to produce more and more enzymes and more efficient enzymes outside the environment. We then compared our enzyme composition by engineered fungus, and we could show that this particular enzyme was having a very competitive biomass hydrolysis efficiency as compared to the most efficient commercial enzymes available in the market. And that was the reason why we could convince our government to scale this particular technology up, and we showed that we could successfully scale it up to 15,000 liter scale, and we produced a good amount of around 100 kgs of enzyme to check the efficiency of the biomass. And also we did techno-economic analysis of the whole process with our partner, which is increase the efficiency. And this is a very recent, just in press, this article came up, where we have characterized the beta glucosidases present in this particular fungus. I mean, this fungus has efficient beta glucosidase production as compared to others, but we didn't know there were like 8 beta glucosidases, as you can see here. We didn't know which beta glucosidases are really making role on the this is a MUG, is basically a specific enzyme activity on the gel. Suggested there are 2 of them which are showing some activity extracellularly. Proteomics showed that there are 3 enzymes that are secreted in more efficiently than other 4. And then eventually, through the deletion of individual enzymes in the genome, we actually could prove that the 2 enzymes out of these 8 are actually making a lot of difference in the beta glucosidase activity. There's a 3A and 3B. We did a lot of structural modeling to understand the features and reason why they were making differences, and these are something that we are going to further engineer and enhance the capability to make it rich with beta glucosidases. This also led to a very good, collaborative project, Indo-UK project, funded by both India and UK government. We actually proven that so these are basically partners that were involved to look at the sugarcane biomass processing. So the sugarcane industry provide electron to LPMO, which needs electron to make the cleavage in the cellulose. So this itself became a new IP that we are trying to valorize. I'll shift to the next goal of making a more intense, dense biofuel molecule. So in fact, butanol is one of them, and you can see that Ali Sami is also probably helping in the organization of the workshop, is authors in a couple of these papers. He worked quite efficiently in our lab for making butanol. So basically the idea was that butanol is very good biofuel molecule, but for some reason because it is being produced by a very strict anaerobe like Clostridium histopalicum, we are not able to valorize or scale up in the industrial scale. So we were trying to make a different strategies, both importing the whole pathway in a much simpler organism like E. coli and trying to see if we can produce more of a butanol, or also we're trying to internally look at whether the fatty acid synthesis pathway, for example, this particular fatty acid synthesis pathway, which normally makes a lot attached with it. So obviously, so this will become a most relevant drop-in fuel. We try to produce the C15, C17 chain length hydrocarbon, which is also called paraffin, basically. And parallelly, because of a very similar pathway, we can also produce a long chain alcohol, which is citral alcohol. And this can also not only used as a fuel, but also in the cosmetic products and lubricants. All our facial cream will have a citral alcohol in it. So basically, we looked at these pathways, so which will take the fatty acid precursor, fatty acyl ACP, and convert it into fatty aldehyde and eventually alkane. And these are the alcohol which can be made correspondingly. We made some discovery here as well. So basically here in a very brief, because of the time, I try to summarize. So this is basically a paper where we have tried to characterize all the freshwater and marine cyanobacteria, which has the ability to produce alkane natively. And then we did some metabolic engineering and discovery of novel aldehyde reductase that can actually convert fatty alcohol. And then we made a metabolic model so that we can engineer E. coli to make more and more long chain alcohol and alkane based on the targets that we achieve. And then this ADO, the final alkane enzymes, yeah, yes, 2 minutes, yes, will be. Funding agency who have been very generously funding us to achieve this. So thank you very much for your attention. Thank you, Professor Zazi, for giving me this opportunity once again. Sure. Yes. So basically, we come. See, at this moment, it is difficult to say commercial value because right now there's no commercial production of 2G ethanol. But when we, try to make some calculation based on what was available in the market, and we realized that it is very, very competitive. So we are hoping that when the 2G ethanol, so eventually what you're going to use is to produce ethanol, right? So you will use this enzyme to hydrolyze biomass, and the biomass will be kind of fermented to ethanol. And we wanted to see if ethanol, when it is made, is it coming in the cost that is, normal molasses fermentation and all the other fermentation that you do is making. So it's slightly higher than that cost. But I think at this moment, we are happy that at least it is not very, very high. It is good enough for the commercial purpose. Sure. I'm sorry. I just saw the chat that there was some problem in the hearing. So I hope I was clear. Yeah. Okay. Thank you very much to all of you. And maybe after some time, I might log out because of my other commitments. I hope it is okay. Bye bye.
Good morning. Yes, yes, good morning, Dr. Azezi and everyone. For some reason, it's not allowing me to share my screen. Do I have restrictions? Do I have restriction in my sharing of screen? Yeah, it says it's telling me that it will not be able to, and if I do until I have to quit. I'm actually in my office at AUC. Do you think I can send you my presentation and you can share it? Oh, now I do. I'm sorry. Now I do. Yes. Okay. All right. All right. Perfect. Um, all right. So, good morning, everyone. Thank you very much, Dr. Azezi and the remaining organizers for inviting me to this event and for hosting this event. Today's talk, I'll be discussing our work in my lab at AUC on the discovery of certain bioactive compounds from extreme environments. The title of my talk is An Environmental Genomic Approach to Drug Discovery and Different Strategy for the Discovery of Novel Genes with Pharmaceutical Applications. The way I will be outlining my presentation, I'll be starting with an introduction on the environment and the extreme environments that we have worked on and our approach, environmental genomics, for those of you who are unfamiliar with those particular approaches. I'll be addressing a few strategies for sample collections and our field work, and then I'll delve into selected environmental genomics materials and methods, including the biogeochemistry of environments, and the discovery of certain anti-cancer and antimicrobial compounds. So these are the different environments that my lab have been looking into. The focus of today's talk are my published work on the Red Sea brine pools. So along the central rift of the Red Sea, there are 25 brine pools, which are deep brine pools that I will be discussing. And this was our first work on extreme environments. Later on, I moved to the West Indies, and we were looking into bioactive compounds from selected volcanoes. What is shown here is Mount Leamagua, which is one of the volcanoes in the West Indies in an island called St. Kitts. We've also looked into selected hydrothermal vents along the Mid-Atlantic Ridge. As I mentioned, the work that I will be discussing today is on the exploration of natural compounds in the Red Sea. As most of you know, the Red Sea has very high salinity, so you would expect that microbes that live in those environments tolerate this high salinity, particularly in the brine pools. The brine pools are along the central rift of the Red Sea, and this is considered extreme environment because they are subjected to the high salinity, the high depth, hence high pressure, as well as other high concentration of heavy metals. So one of the brine pools that we looked into is at a depth of 2,200 meters below the surface of the sea water. It is hot. The temperature is 72 degrees Celsius. It has very high concentration of toxic gases as H2S and very high concentration of heavy metals, as I will show you in a little bit. Hence, the organisms that live in those environments are known to be extremophiles, or organisms that love extreme environments. So one of the first brine pools that we went to is shown here. This is between Saudi Arabia and Sudan. These are known as the Discovery Deep and the Atlantis II deep brine pools. This shows a closer image of these two brine pools, which are in close proximity. This is the Atlantis II, which is at a depth of 2,200 meters below the surface of the seawater. It's hot with a temperature of 70 degrees Celsius, saturated salt up to 250 PSU, and very high concentration of heavy metal, in addition to the fact that it's an anoxic environment. For those of you who are familiar with like the Mediterranean brine pools, you would know that the Mediterranean brine pools are, first of all, not so deep, and also it's not subjected to different harsh physico-chemical conditions. The Discovery Deep that is adjacent to the Atlantis II Deep is also a harsh environment, but it doesn't have as much heavy metals as you'll see in a few minutes in comparison to the Atlantis II. We've also collected samples from an adjacent site that we call it non-brine site, and we believed at the time that these sites are not an extreme environment, and so we can use it as a control for our studies. So if we take a look at the temperature of one of the brine pools, and this graph shows the temperature when compared to depth. So once you hit the brine pool, this is at around 1,980 meters below the surface of the seawater, you will see that the temperature starts going up. And once it hits a little bit above 2,000, it reaches 50 degrees Celsius. And the deepest part at the time that we collected was a bit over 68 degrees Celsius. So this site is an exotic site that we, as well as other scientists, have been looking into, and it's referred to as the LCL, or the lowest convective layer. This is an isolated area, and obviously organisms or microorganisms, because these are the only organisms that have been discovered to date that survive in the lower convective layer of this Atlantis to Red Sea brine pools, are the extremophiles that we are interested in. So what we've been doing is we've been collecting DNA and RNA from these environments. And then we subject those DNA to sequencing, and sequencing of the DNA would lead to the production of reads. And those small reads that are the output of our sequencing would require a lot of bioinformatic analysis for us to take a closer look at the whole picture. So environmental genomics is basically putting together pieces of the puzzle or pieces of the read to end up with an entire genome that then we can analyze and look into. So after we do this assembly of the reads, we would then be interested in a particular gene that has been previously known to have certain bioactivity. And these are the ones that we'd be interested in to explore for the discovery of drugs or enzymes that can be used in biotechnology. There's another approach which is known as functional genomics, and this entails the construction of a phosphate library. And in this case, we will be looking for a very specific function that we can then look for the bioactive compound. So this was my introduction into the environment that we've looked into and our approach, which is environmental genomics. The next thing I will be discussing are the field work. And the field work that we need to do entails collecting samples. This is one of the research vessels, which is the Oceanus from Woods Hole Oceanographic Institute, where we go on to collect the samples. And we collect both sediments and water samples. So this is a sediment core, one of our sediment cores. This is 3.5 meter core. So once this core goes in the water, it accelerates when it's close to the seabed, and then it will end up collecting a 3.5 meter sediments that then comes up on top of the research vessel for further analysis. Another sampling device that we use, but in this case we use it to collect the water, is termed CTD. CTD is an acronym for conductivity, temperature, and depth. This is one of the CTDs which was on one of our research vessels, and each bottle is termed the Niskin bottle. This Niskin bottle is actually open. And then once we reach a certain depth or a certain pH, because we're monitoring this on deck, we would then close the Niskin bottles, and then it would come up to us with the water from this particular site. So, for example, if we take a look at the sediments that we collected, this is a diagrammatic illustration of the 3.5 meter core from both the Atlantis 2 and the Discovery Deep, which are the two brine pools that I described to you earlier. And if you just take a look at those two sediment cores, you can definitely see that both cores have different or distinct lithologic characteristics. And therefore, you would imagine that the organisms that are likely to survive in this environment would be different. And when we looked into the carbon, hydrogen, nitrogen, and sulfur profile in these sediments, you can see that there are different sections of these sediments, and particularly the deepest part of these sediments have unique, for example, with Atlantis 2, has unique and very high sulfur content, where in the Discovery Deep, it had the highest nitrogen contents. And this allowed us then to predict that the organisms and the genes that we're likely to discover in those sites would have tolerance for either the high sulfur or the high nitrogen that is present in those sites. What I'm showing you here is a chemical profile, an enrichment plot. This enrichment plot is normalized to aluminum. Unfortunately, here where it says undefined, this is supposed to be metals. And I know that this metal here is the aluminum because aluminum is the reference sample, and any other metal above aluminum is enriched in this sample. So up here, this red arrow points to mercury, and mercury is very high in the Atlantis II brine pool, and allowed us to discover a lot of mercury detoxifying enzymes from these pools that can be used in bioremediation. We can also present the chemical profile using a hierarchical clustering to look at the different heavy metals in both Atlantis II and the Discovery Deep. So this heat map shows the relative abundance of metals in the Atlantis II and the different sections of the Atlantis II versus the different section in the Discovery Deep. Here, I don't know if you can see it clearly, but here are the different metals. So for example, mercury, which is here, you can see that it's extremely abundant in the Atlantis II, which is not the case in the Discovery Deep. So with this, I've shown the sampling collection and the different biogeochemistry in two different environments that we looked at. So after we collect the DNA from these environments and we subject it to sequencing, we can have two approaches. Approach number one is termed a structural genomic approach, where we basically just do shotgun sequencing with extensive bioinformatic analysis and look for specific genes that can have biotechnology or pharmaceutical application. The other approach is termed a functional genomic approach, where we construct phosmid libraries. And in this example, we would have around, for each library, would be between 10 to 20,000 clones that we can further functionally screen them. And once they're functionally screened, we can select this clone and investigate further what it is doing in this environment. So the first approach that I will be discussing is the structural genomics, which is the shotgun sequencing that is followed by the extensive bioinformatic analysis. And once you've identified a gene that looks exotic, let's say it's an antibiotic resistant gene, which is what we've done. So this gene, you can synthesize it, you can clone it in a vector, you can express it, and then you can look for the biochemical characteristic of this gene and its extremozyme potential. And indeed, one of my former PhD students in my lab has looked into antibiotic resistant gene from the Red Sea brine pool and has found two genes that he thought was quite exotic. One was an aminoglycoside phosphotransferase, and the other was a beta lactamase. And this was done following sequencing of the Red Sea Atlantis II brine pool. One of these aminoglycoside phosphotransferase only had 58% identity, and the beta lactamase has 55% identity. And indeed, he published this work where the beta lactamase was extremely heat tolerant and was therefore can be used in the plasmids that can be cloned in extremophiles or organisms that are subjected to high temperature during the expression of the protein. So this was the shotgun sequencing. The other approach that we used is the phosmid library, which is the functional screening or functional genomics. And what I will be showing you is an antimicrobial assessment of the phosmid library. And once we identify the clone, that is resistant to a certain or can kill a certain bacteria, then we can extract this clone and further assess its enzymatic property. And so this is the materials and methods that we use in my lab, which is an environmental genomic, both a structural and a functional environmental genomic approach. So let me show you some data that we have from both the shotgun sequencing, the structural genomics, and the functional genomic approach. For example, Layla Zico in my lab, when she looked into the Red Sea brine pool metabolome, she was interested in bioactive compounds, particularly those with specialized metabolism genes. And she compared between two brine pools. One of them I haven't had the opportunity to talk to you about, which is the Kabrit Deep. And this Kabrit Deep brine pool, as you can see, has a very different metabolome just by looking at this Venn diagram from the Atlantis 2 brine pool. And you can see that in the Kabrit Deep, there are non-ribosomal peptide synthetases with different chemistries in the Kabrit versus the Atlantis. We've also identified polyketides, which are produced by many organisms, particularly filamentous fungi. And a lot of these polyketides are known to be clinically important with antimicrobial, anti-cancer, and immunosuppressive properties. But we've also identified in this Kabrit Deep microscenes, which are antibiotic peptides that can block vital functions and can create pores in bacterial membranes, and hence can be used as an antimicrobial. Now, when we looked into the chemistries of 5 core structures that were predicted by a certain pipeline called anti-SMASH, including non-ribosomal peptides and polyketides, you can see that in the Kabrit Deep and the Atlantis 2, we've identified novel and different non-ribosomal peptides and polyketides that can further be investigated for their different bioactive compound properties. Now, with the functional genomic approach, when we screen the clones, this is an antimicrobial screening. You can see that this clone managed to kill this bacteria. So, and this one as well. So then what we do is that we would take this clone and further investigate it. And one thing we did is that we did a cell viability assay, which is an MTT assay, to look for the effect of this clone on certain cell lines, including the MCF7, which is a breast cancer adenocarcinoma. And indeed, it led to diminishing this the viability of those cells. We've also used the U2OS, which is a bone osteosarcoma mammalian cell line, and the human telomerase reverse transcriptase immortalized cells, which didn't have that much effect. This shows 2 different clones. As you can see, the color in red, which is one of the clones that has extensive cytotoxicity on all the cell lines. On the other hand, the green clone only shows cytotoxicity and significant cytotoxicity with the breast adenocarcinoma. So therefore, there were selected clones that would have antimicrobial and selected clones that would have anti-cancer activities. Then what we do is that we would take, this is one of the clones that we sequence, and we would annotate those clones. And you can see, for example, in pink here are the genes that we are interested in, because the one in red is the NRP, and the one in pink is the polyketides, which are the ones that are known to have bioactivity and can have applications in anti-cancer discovery. So this work that I presented was started by Layla Zico in my lab and was continued by Marina Nabil, where they have looked into those anti-cancer and antibacterial effect of selected clones and looked at their adaptation to the environment. So with this, we've looked into extreme environment. Yes. Sure. We've looked into extreme environments, microbiome and metabolome. We've looked at genomic data and we've established a genomic data base. We're still working on the establishment of this genomic database, not just from the brine pools, but also from other extreme environments, particularly in the West Indies. And we're constructing phosmid libraries from these extreme environments that could be functionally screened for natural compounds and the characterization of interesting natural compounds that can be used in biotechnology and pharmaceutics. So this is my team. We train them young, so some of them are very young. No, these are actually the kids of members of my team in one of our gatherings. The work was supported, that I've shown you, by the British Ecological Society and by the School of Science and Engineering Research, Dean Research Grant, and the University of Medicine and Health Sciences in the West Indies. And this was done in collaboration with the Technical University of Berlin and Dr. Rami Karamaziz at Cairo University and the Cancer Hospital in Egypt. I thank you very much for your attention. Yes, Dr. Azizi. Actually, a lot of the anti-cancer compounds and a lot of the antibacterial compounds that are on the market have been identified from extreme environments. Unfortunately, I do not have the slide for that. Maybe this is definitely was one of the slides that I had in previous conferences that showed the percentage, and it's not over 50%, but at least within 20% range, if I remember correctly, have been commercialized. Mm-hmm. Thank you very much.
Hello, السلام عليكم ورحمة الله وبركاته. Sound okay? Okay. Just I will share my slide. Okay. Okay. Hello. Yeah, okay. Good morning. I'm Heba, lecturer at Biochemistry Department, Faculty of Science, Zagazig University. First, I to thank the organizing committee for giving me a chance to present my research work here in such informative and organized conference. My title talk is Water-Soluble Polymer Mediated Efficient Delivery of CRISPR-Cas9 for Targeted Gene Therapy of Cancer. Outlines concept of gene therapy, nuclease-mediated genome editing, CRISPR-Cas9 system, lung cancer, and epidermal growth factor receptor, classification of delivery methods, aim of the work includes design of a CRISPR-Cas9 system for EGFR editing, and development of a novel drug delivery system for efficient genome editing, mean achievement and results, conclusion, and acknowledgement. Firstly, I would like just to give short talk about what is the concept of the gene therapy. Gene therapy is based on the principle of genetic modification of the living cell for treating various disorders, and the final goal of the gene therapy is to cure patients who suffer from genetic disorder, including congenital diseases, infectious disease, and cancer. As shown here in this figure, this cell with non-functioning gene, and after adding DNA containing functional version of the lost gene, the gene started to work again. Genome editing is a type of gene therapy, and it will be challenging therapeutic tool in the future. Genome editing depends on one of these 4 artificial nucleases: meganuclease, zinc finger nuclease, TALEN transcription activator-like effector nuclease, and CRISPR-Cas9 system clustered regular interest-based palindromic repeat. Each of these enzymes consists of 2 domains: specific DNA binding domain here, linked to protein with non-specific cleavage property. Genome editing is induced by a double-strand break within the genome. This double-strand break can be repaired by 2 pathways: non-homology end joining and homology-directed repair. The non-homology end joining for random insertion or deletion within the cleavage site, which lead to frame shift mutation, and homology-directed repair, which use DNA template for the precise correction of the cleavage site. Among these enzymes, the newest genome editing tool is the CRISPR-Cas9 system, and is the most investigated system and has been applied in clinical trials of human cancer. What is the CRISPR-Cas9 system? CRISPR-Cas9 system consists of 2 parts: Cas9 protein and guide RNA. Cas9 protein act like a pair of scissors to cleave DNA. Endonuclease protein consists of 2 nuclease domain: RUVC domain and HNH domain. Guide RNA here consists of 2 changeable part and fixed part, which is called transactivating RNA. So this fixed part not changed at all, but the changeable part contains conserved repeated sequence and a spacer of 20 nucleotide guides. This spacer can be redesigned to target any sequence within the genome. So the key of the CRISPR-Cas9 is here with this 20 nucleotide guide RNA. So we can redesign it and start to use. However, this nucleotide or this spacer should be adjacent to 3 base sequence called BAM, spacer adjacent motif. It can be, for example, NGG. And why? Because this BAM sequence is very important for the activity of Cas9 to cleave the DNA. On the other hand, here is also the mechanism of how the CRISPR-Cas9 work, and also as shown here in both figures, after the CRISPR-Cas9 recognize the specific target within the genome through this spacer, so it can bind to the complementary DNA strand within the genome and start to induce double-strand break by which domain of Cas protein by HNH motif, and while the RUVC domain can cleave the non-complementary strand. Just here, I would like to show why I target dermal growth factor receptor and what is the dermal growth factor receptor. Lung cancer is one of the most commonly diagnosed malignant type of cancer. Dermal growth factor receptor belongs to ERBB family. So there are 4 types of ERBB. EGFR is ERBB1, and its family of receptor tyrosine kinase and exerts critical functions in epithelial cell physiology, and it's overexpressed in different types of cancer, especially lung and breast cancer. This EGFR is working well as proto-oncogene in the normal cell, but when there is mutation, EGFR oncogene start to be overexpressed and start to be a key driver of the cell proliferation, differentiation, migration, and angiogenesis, contributing to the lung cancer. So EGFR provides cancer cell with a survival advantage and resistance to the therapy as well. So targeting the EGFR offers a window of opportunity in cancer treatment. CRISPR-Cas9 system was used here to target dermal growth factor receptor oncogene, which is promising molecular tool for lung cancer treatment. And as I said before, it's a driver of the cell proliferation. The success of the CRISPR-Cas9 system is influenced by the effectivity of the delivery system used to carry it into the cells. And as shown here in the figure, the delivery system classified into 2 types, viral vector and non-viral vectors. The non-viral vectors include physical method and chemical method. Among these vectors, chemical methods are being considered to be the most promising one. Aim of the work: design of CRISPR-Cas9 system for cancer gene therapy through knocking out epidermal growth factor receptor oncogene and studying the most efficient CRISPR-Cas9 target. Introducing a novel drug delivery system for increasing the efficiency of our CRISPR-Cas9 system construct. This schematic demonstrates the main objectives of my research. As I said, construction firstly of the CRISPR-Cas9 for targeting epidermal growth factor receptor gene within the chromosome 7 and loading it into the a novel water-soluble lipopolymer as the novel delivery system for the genome editing. Firstly, I will talk about the design of CRISPR-Cas9 system for cancer gene therapy through targeting epidermal growth factor receptor. CRISPR-Cas9 system plasmid used in this study was obtained from Agen, and it contains Cas9 protein, guide RNA scaffold, and coding gene for monitoring the plasmid inside the cells. The first step in my experiment was the digestion of the plasmid by specific restriction enzyme called BBS1 into a linear form. And to monitor the success of the digestion process, I performed agrogel electrophoresis, and as shown in the figure, the plasmid converted to the linear form. CRISPR-Cas9 plasmid was used, as I said, to target epidermal growth factor receptor oncogene, which is a promising molecular target of the lung cancer treatment because it's the driver of the cell proliferation. The guide RNA sequences were designed by the CRISPR-Met tool. So this is one tool of the different tools used to design guide RNA. For example, another tools like blanching tool, but in my study we used CRISPR-Met tool to target selected exons as shown in this table. These exons 1 and 19 and 20, 21 were chosen specifically due to their role in EGFR encoding function. The design guide RNA sequences were cloned into the cleaved plasmid, ligated and multiplied in Escherichia coli in LB medium, and this new plasmid will provide in a high amount to be used in our cell culture experiment. The success of the insertion of the guide RNA, which will target the epidermal growth factor receptor into the CRISPR-Cas9 plasmid, was monitored by DNA sequencing, and as shown in these electrophoregrams here, which shows the insertion of some guide RNA here within the sequence. Our variable genome editing, as I said, there are 2 two methods or two mechanisms of the genome editing. Our variable genome editing will be non-homologous joining, which will lead to frame shift mutation, and by this, the oncogene can be disrupted. EGFR knockout was detected by sequencing the genomic DNA and quantitative real-time PCR of the extracted RNA. After extracting the genomic DNA, performing the transfection experiment with our CRISPR-Cas9 system and extracting DNA and RNA, started to analyze my results. We found that according to codon code aligner tool, I used it to compare between the sequence of the control and sequence of treated cells. And as shown here, the top sequence represents the control without any noisy or any gene knockout, and the bottom sequence shows slight increase in the background noise. Also, I used type to detect the percentage of mutation efficiency by detecting the possible insertion and deletion within the genome sequence. And according to my results, type tool showed that the mutation efficiency in the case of exon 17 of epidermal growth factor receptor was 7.5% after 48-hour incubation period. And by detection of the EGFR mRNA expression level by real-time PCR, the CRISPR-Cas9 system targeting epidermal growth factor receptor exon 1 and exon 17 and induced a 20% decrease in EGFR mRNA expression level after 48-hour incubation period. Summary of this aim. In this study, we demonstrated a novel approach to destroy the oncogenic EGFR mutation in lung cancer using CRISPR-Cas9 through designing guide RNA targeting different exons of epidermal growth factor receptor. Mutation efficiency of proved to be 7.5 after 48-hour incubation period. So further improvement are needed to enhance the gene therapy efficiency by CRISPR-Cas9 system. The second aim is introducing a novel drug delivery system for increasing the efficiency of our CRISPR-Cas9 construct. Clinical applications of CRISPR-Cas9 are hampered by their inability to reach the intended target tissue through the cell membrane and exert this therapeutic activity in vivo. For this reason, we have decided to encapsulate our CRISPR-Cas9 construct into a novel cationic water-soluble lipopolymer. This water-soluble lipopolymer was prepared or synthesized by two main agents, polyethylene and cholesterol chloroformate. This schematic figure here, which represents the formation of water-soluble lipopolymer complex and its and the condensation of the plasmid DNA into a water-soluble lipopolymer to form water-soluble lipopolymer plasmid DNA complex. The condensation here or loading here depends on the electrostatic attraction between the negative charge of the plasmid DNA and positive charge of cationic water-soluble lipopolymer. To detect the molecular weight of our synthesized water-soluble lipopolymer, we have decided to detect the specific charge, and the specific charge was determined by the titration of sodium didosyl sulfate by polyethylene and water-soluble lipopolymer. Taking the charge neutralization point, it will demonstrate that specific charge had decreased in the concentration stage. So the ratio, so as you see, the specific charge of water-soluble lipopolymer is less than quantitative specific charge of polyethylene, and this means that the ratio of the specific charge for both polymer is 0.44, meaning that the water-soluble lipopolymer has 56% less specific charge. And since the specific charge as shown in this equation depends on the molecular weight of the polymer and number of charge in the single molecule, so that the decreasing of the specific charge of the water-soluble polymer make your dose because increase of the molecular weight and decreasing the number of protonated nitrogen of polyethylene or of water-soluble polymer. This provided a chance to estimate the molecular weight of water-soluble polymer, and after that, we started to make some characterization of our prepared water-soluble polymer, and it was the water-soluble polymer DNA complexes was prepared by changing the ratio between the polymer and DNA. We kept the amount of DNA constant, while the MB ratio was increased from 1.25 to 20. So the changing was in the amount of the polymer used. So I mixed a different amount of water-soluble polymer to constant amount of DNA. And the encapsulation after that was examined by using garage gel electrophoresis, as shown here, which shows that the absence of the DNA band from 5:1 ratio. This polymer started, I characterized it by light scattering to determine the particle size and zeta potential as well. And as shown in the tables here, I used two plasmids in this study as control. So our construct BX458 plasmid CRISPR-Cas9 and BGV-N3 such control or as control plasmid in our study for comparison only. And as shown here, the size started to increase by adding water-soluble polymer, and also the charge started to be more positive after adding the water-soluble polymer to the DNA. After that, these water-soluble polymer DNA complexes were characterized by scanning electron microscope and transmission electron microscope. And as shown in these figures here, for example, A and B shows images of water-soluble polymer BGV-N3 plasmid using 5:1 ratio preparation under different magnification. See just the water-soluble polymer itself without DNA, and D and E and F images showed the complexes of DNA with the water-soluble polymer from 10:1 ratio preparation. Before starting to use the novel cationic water-soluble polymer as a carrier for CRISPR-Cas9 system, we performed the cytotoxicity assay firstly to check the cytotoxicity of this prepared polymer. And the cytotoxicity of this polymer and also its DNA complexes was measured on A549 cell line, lung cancer cell line, by MTT-assay. According to our results, the polymer is highly toxic to the cell at 304 nanogram, as shown here. So the cell viability decreased significantly upon the increase of water-soluble polymer concentration, which indicate remarkable dose dependence of cytotoxicity. As shown, the presence of DNA plasmid DNA didn't increase the cytotoxicity of the system. And also, we compared the cytotoxicity of the water-soluble polymer with polyisonomine and the traditional one, lipofectamine 3000. And according to our results, polyisonomine is extremely toxic to the cells, as shown here, with the same amount, 104, is very comparable for the water-soluble lipopolymer after 24-hour incubation and 48-hour incubation as well. So the resulted water-soluble lipopolymer is very safe at low concentration or low amount for using a transfection agent. What's the transfection? Transfection is a process of plasmid DNA uptake by the cells. The efficiency of this process using the water-soluble polymer DNA complexes at various MB ratio. I know that there is a ratio which is not toxic, but I tried also to use different ratio to see the difference between the transfection efficiency of all of them. And we determine the transfection efficiency through the green fluorescence of the expressed EGFB protein. As known, this CRISPR plasmid contain EGFB gene coding. And according to the results of the flow cytometry, here the most efficient transfection was achieved at 20:1 MB ratio. And the transfection also was verified by using a fluorescence microscope, and which in the GFB expression is more intense in cell transfected with the water-soluble polymer plasmid DNA than the polyisonomine plasmid DNA complexes at 10:1 ratio. So from here, we decided to use water-soluble polymer DNA complexes 10:1 ratio as a transfection reagent for our CRISPR-Cas9 system. And also, we monitoring the GFP expression by using confocal microscope. And after transfection, the cells, the nuclei of A549 cells, lung cancer cell line, were stained with host fluorescence dye, and the results reveal that more EGF is expressed using water-soluble polymer as a transfection reagent than polyethylene amine. Yes? Okay. In the other words, water-soluble polymer complexes showed higher intracellular distribution than polyethylene amine DNA complex. After transfection efficiency was achieved using the water-soluble polymer carrier, the genome editing of the designed CRISPR-Cas9 system was monitored, and also for demonstrating the biological activity of water-soluble polymer BX458 complexes at 10:1 ratio. But according to TIDE results, it showed 8% efficiency, but contrary to real-time PCR downregulation of EGFR gene, or sorry, the knocking out reached 85% here. So this demonstrates the visibility of applying cationic water-soluble polymer in combination with genome editing materials, which could broaden the application of nanomaterials in gene therapy. Water-soluble polymer cationic lipopolymer was synthesized as a novel drug delivery system to improve the efficiency of artificial nuclease action in the cells. The specific charge was determined by saturation of sodium sulfate, and water-soluble polymer and water-soluble polymer DNA adducts didn't show significant toxicity at concentration of 150 nanogram per well. Also, it proved that water-soluble polymer can be used as transfection reagent. Knocking out of EGFR of our CRISPR-Cas9 construct was improved. And thank you. And finally, I would like to express my deepest appreciation for the organizing committee for such a well-organized and informative conference, and would like also to acknowledge the funding for my research, and especially the cultural affairs and mission sector in Egypt. Thank you very much. Thank you. Okay.
Yes, sir, Professor Azad. Thank you. Thank you very much for your introduction, clear introduction. Actually, it's my pleasure to be part of this eminent scientific symposium, and also to be among these eminent scientific community. And thank you for your kind invitation, and also to present my talk today. So I hope that the sound is clear and everything, and that also the shared material is also on the screen. So you can hear me, right? Hello? Excellent. Excellent. Thank you very much. Okay. So simply today, the title of my talk is Benchmarking in Structural Bioinformatics, Recent Applications Using Decoys 2.0, actually from the title. So I will simply deviate a little bit from the mainstream of the experimental area. So I'm going to theoretical areas. So I'm talking about bioinformatics and cheminformatic approaches. So the important keyword here today is about benchmarking. And here, highlighting benchmarking, actually we will just simply present the recent application of one of these benchmarking sets. We call it Decoys 2.0. So the outline is quite simple. I will simply dive a little bit on the concept of benchmarking, and then I will move into the application of benchmarking. So starting with the concept of benchmarking. So benchmarking simply is a very important procedure in the virtual screening. And actually we use it to enhance the efficiency of virtual screening. And as you know, simply virtual screening means that you have a digital library of compounds that you try to screen them against a certain biological target to treat certain diseases, for example, in a drug discovery context. And usually you use what's so-called screening protocol like docking, like pharmacophore, and all these kind of stuff. Actually, molecular docking is among the frequently used screening protocol for virtual screening. And at the end, actually, you are aiming to find certain hits. So hits meaning that you are actually predicting certain compounds screened from the digital library that are able to bind to a certain molecular target. And this is actually predicted theoretically. And of course, the subsequent step is trying to validate that in an experimental way. Actually, the problem with the virtual screening itself is the false positive rate. Actually, most of the hits that actually you gain from the docking protocol are not active. They are artifacts. Most of the time, they are false positive. According to recent reports, they are simply, their success rate does not exceed even 1%. So that's why benchmarking is trying to elevate the success rate, trying to minimize the false positive rate. And here, actually, we need to understand, so what is benchmarking? Benchmarking simply is an evaluation of the ranking performance. So what we do, simply, we have certain actives. So we know previously there's some, for example, if you're trying to make a drug discovery campaign against COVID-19, for example, or cancer, or whatever. So you collect certain actives, known drugs, known inhibitors for the enzyme, or known binders to the protein of interest, and then you try to compile certain decoys. Decoys mean there are compounds similar to the active, but they are not active. And then you blend them together and you're trying to deceive the docking tool, the docking protocol that you're using prior to a real virtual screening campaign. So what simply you will do, you will simply perform a virtual screening for the actives and decoys. This is actually a benchmark set. So you have active and decoy, we call them benchmark set together. And then you will see a ranking performance. So the ranking is coming, depends on the protocol you're using. For example, if you're using docking, so you have a score, and simply you order your list according to the score. And in the ideal life scenario, what you will simply get, you will get, okay, I'm using docking of true actives against false actives, false positives, and we should expect that the active will show the best rank, the best score, 1, 2, 3, 4, 5, if you're starting with 5 actives. And this is actually in the ideal life scenario. And you can simply transfer this data, transfer these results into statistical form, like we use ROC curve, receiver operating characteristics. They're very well known, but we use the logarithmic scale. We call it P, like PIC50, PKD, PKI. So we're trying to mimic the biological or the affinity data at the end. So in this kind of ideal life scenario, what you will see, you have in the x-axis, you have the false positive rates reflecting the decoys. You have the y-axis, the true positive rate reflecting the actives. And if you have, at the early enrichment, you have the best and priority, the highest priority of the scores coming from the active, you will get one step forward into the y-axis toward the true positive rate. And if you have all the active reached at the early beginning, you will have all you will recognize the straight line coming upwards toward the y-axis until you finish all the true positive of the active, which is 1 here representing 100% of all your actives. And then your program will simply see the ranking coming from the decoys afterwards. And in this way, you can see here the area under the curve. You can assess it according to the performance. Actually, in the real life performance, you will get something like that. So you will have your actives, and it should be simply scattered, something like that, because simply the docking tool is not perfect. And also at the same time, the actives themselves actually, the decoy themselves also, they be quite challenging. And in this case, you will simply see that you have here, for example, you have certain curves like that. And this curve simply will give you a differential assessment between docking tools. For example, if you have more area under the curve, that means you have more active at the enrichment, and you can use this protocol and use this simple metric to compare between actives and decoys and to compare between the docking tools. And one practical example is this one. For example, what we really see, this is one of our papers in the very beginning about the decoys. You can really see here we benchmark different docking tools. I'm quite sure that a lot of working groups, for example, using MOE, for example, like in Alexandria University, like in Cairo University, like in GUC, for example, and many others use other different tools. And the question is whether MOE or whether the gold or whether any other tool is really efficient for your task. Is it really can be used for visual screening in your protein? And this is actually actually show the importance of benchmarking. And you can really see here with this color code, color line, you can simply perform here, as you really see, the different benchmarking. So here, for a certain target, we apply active and decoys, and you can really see the area under the curve. The very important is to look at the early enrichment, and that's why we use the logarithmic scale, so you can really enrich here. You can really have a zoom in the early area under the curve here at the very beginning of the cutoff, you see. And this is quite important because this is for the pharma industry, means money. So if you have a docking tool that can really recognize actives at the very beginning, that means it can really see in the very beginning. If you, for example, screen a library of 1,000 compounds and you have a cutoff at a 1%, then you are really interested in only 10. So in this case, you can say, okay, I will use it in a real-time versus screening campaign. I will screen 1,000 compounds. I will only purchase 10. I will only test 10. So in this case, I simply save 90% of the operation cost and and also I would save 90% of the total cost of the drug discovery here in the very early beginning. So that's very important. It means money. So if you have a very efficient tool that can really do this, you can rely on it for the real virtual screening campaign. And does it only mean reaching area of the curve? This is a very important question, because if I do a thought experiment very fast with you, you will simply say, okay, maybe the decoys are very easy. So in this case, if the decoys are very easy compared to the actives, so any docking tool can simply differentiate between the actives and decoys and will simply grant higher score for the actives compared to the decoy. And that's actually, here we have an example of 5 compounds. And the question is, can you find 2 actives? And you can really see from the answer that you have A and C, that both are HIV proteases, and simply they have the same topology. So they have the same chemical features. They are amide or carbamate or whatever amidic linkage incorporated. You have here in the same, so it's you have here like a urea derivative. They have certain number of aromatic rings, certain number of heavy atoms, and etc. So A and C look very similar. While, for example, E, this is a progesterone receptor antagonist, so it's a steroidal nucleus. You have here D, it's an anti-influenza drug, so it's a sugar-like molecule. You have B, for example, it's a 5-HT agonist. So simply what you can really say, you don't need, in this case, a docking tool. You can even do it with your eyes. And here comes the question. If you have any docking tool and you try to benchmark with this particular benchmark set, it will give you a misleading results, because A and C are very similar, and B and the decoys, which you have B, D, and E, are very easy. So whatever you have enrichment, whatever you have area under curve, does not really reflect a good performance. And here actually comes the question. So what we make it challenging? And just to take care, we need to make it challenging somehow in order to also avoid the activity intrinsic properties. For example, if you have here 5 compounds, if you just think a little bit, if you have time, I would make something like a discussion with the audience, but of course we don't have time now. So simply, if you have look at that, you can really say, okay, what are the two actives? And if you just look further and further, you will say, okay, probably they have the same features, probably they're really close to each other, so it's more and more challenging. And if you have a docking tool that can simply differentiate and find the actives, granting them a high score compared to the decoys, in this case, you can trust it, you can use it for a real live virtual screening, and you may trust also a higher success rate for the virtual screening. And here, actually, the answer, you will find that ABC are decoys, only E and D are the actives, and they all look similar. So if you have a docking tool that can really differentiate, in this case, that would be interesting. And here, actually, the answer. So decoys 2.0, this is an automatic workflow based on artificial intelligence. The keyword is immediate neighborhood classification. So we developed that, actually, and you can really see more details about that in the website. So we have a couple of papers about it, so how it works. And the main mission for decoys is simply to develop and to mine decoys from the provided active. So for example, you are a user and now you're interested in a cancer drug discovery using docking or virtual screening. So you provide decoys with the actives, and then it will simply mine to you. It will simply provide you with the decoys, and then you can use it in your work for benchmarking. And then you can, afterwards, you can decide which docking tool you can use it, for example. So this is actually the idea of Decoys 2.0. And if you visit the website, you will find a lot of features. Just here, the important is that it covers the target space. So it covers around 80 diverse targets from the target space from the protein data bank. And each, we have an active set composed of 40 actives and also around 1,200 decoys. So you can use it in that. So the question is, what are other applications? So this is actually the concept now. We understand the concept of benchmarking. We need to use it to evaluate our docking tools. So the question is how we can use it afterwards. Are there any other recent applications that we can use? Actually, yes. This actually review article simply nominated that decoys is one of the best state-of-art benchmark sets actually developed, and also subsequent reports also validate, confirm this finding. So the application of benchmarking, simply, I will give you a couple of examples, one in the area of artificial intelligence, and then I will move quickly into the area of COVID-19 drug discovery. So in the area of artificial intelligence, here I will move into the other working group work, because it's important for us not only to see our own work, but also to see the conception and also how other working groups can conceive our work and can use it for further studies. So here is a gold standard data set for virtual screening that says simply you need to train and to test a computational method. You need a benchmark set, and also you need a benchmarking to compare performances. And this is actually the guiding sentences for us. So the example is for vScreenML. This is a scoring function based on machine learning, which is a type of artificial intelligence. So they call it vScreenML. And actually, the working group from University of Kansas, what they simply did, they developed a machine learning classification that can reduce false positives in structure-based virtual screening. And why this specific article? We have many articles we can really talk about, but why this specifically? Because they used benchmarking not only in the validation or testing, but also they use it in developing. So they need to develop a data set and to train it. So they use it actually in the very beginning. And the idea here, just to make the long story short, they simply said, okay, we will go to the protein data bank. We will collect all protein data bank complexes with ligands, and then simply they generated decoys from other providers like .e, and then they simply try to generate and compare the scores and collected some features and descriptors in the binding site coming from both the true binders and also from the generated decoys. And afterwards, they try to train it. How they train it? Simply, they have, you can see here from the x-axis, you have the scoring scheme they used, and you have the y-axis, the frequency, and you can really see two populations. The population of the actives are the blue colored, the population of the decoys are the red colored. And if you are able to fine-tune the scoring scheme, the basic one, which is Rosetta, which is a very popular tool they use, then you try to incorporate more and more like artificial intelligence scoring function like SGBoost. It's a machine learning algorithm. So what they simply do, they try to discriminate between the active and decoys, and you can really see that the blue color and the blue population and the red population are really discriminated by time. And after final optimization, they they simply develop and announce vScreenML. So this is just showing you how they train it based on the concept of actives and decoys and based on the concept of benchmarking. And what's really important for us that they used for a final decision and also for validation for their approach, they use decoys 2.0. So how they do it actually? So they did it simply by comparing the vScreenML compared to other state-of-art artificial intelligence scoring functions, something like NN score, something like random forest score, random forest score, RF, and others. And you can really see here the idea is simply that you have, if you can see more green dots towards the vScreenML, that means you have higher enrichment factor, so you can really see more and more actives at the early enrichment. And you can really see that the vScreenML simply, which is developed based on benchmarking and now validated against decoys, shows and outperforms the other scoring functions. So they use it and they say, okay, based on the results of benchmarking against decoy 2.0, we now announce it officially, and they simply used it for a prospective virtual screening and experimental validation. And for the experimental validation, they used a simple a simple CAT, which is in vitro enzymatic assay against acetylcholinesterase enzyme, which is available, which is not expensive. So they used it, and they used vScreenML for scoring. And then they say, okay, now we have different compounds, and you really see from the y-axis here, you have the percentage of acetylcholinesterase activity. You will find this compound, AC6, for example, that shows the best inhibitory activity against the enzyme. And that's why they used it, and they just simply made the experimental work, and they got the IC50, and at the end, they also could crystallize it with the enzyme. So this is a long process they did, but actually I tried to make it short and to see how very efficient benchmarking can be used in scoring, in developing and scoring, and in validating the scoring function. Another example just has been recently published in the Journal of Advanced Research with other group in Spain, and UK, and USA. Also, they develop a so-called S-score, which is a scoring function based on also machine learning classification. And they mainly use decoy 2.0 in order to validate and also to develop the scoring function. So now moving quickly from the AI into the COVID-19, and the COVID-19 drug discovery, as you know, it has been started since a couple of years ago. And from the very beginning, we tried to contribute with the benchmarking idea to the community, because a lot of people using just theoretical work like virtual screening, undocking, and try. So we tried here to provide an example for that, a couple of examples actually. So here we talking about certain target, which is called SARS-CoV-2, peptide-like protease, one of the enzymes, or one of the important proteases for the virus. And we try to, our motivation is to provide a benchmarking study, because there is no benchmarking study at that time. So we have a couple of challenges. The first challenge, we don't have a valid model. We have BDB structures, but it's not a valid model because it does not reflect the binding situation for small molecules. So we modeled it ourselves. And then afterwards, we need to generate decoys 2.0. So we need actives in order to generate decoys, but we didn't have. So we went to the similarity criteria. So we went to the ancestor of the COVID SARS-CoV-2, peptide-like protease. We went to the ancestor, which is SARS-CoV-1. And we performed what's so-called cross-benchmarking. And here's simply, you can really see from these graphs, you have three colors. You have three docking tools, Fred, Vina, and plants. And you can really see one of these colors show the highest performance. That means it shows the best area under the curve. And we say, okay, based on that, we can use it. And we recommend it for further screening of further virtual screening campaigns. And to get more insights about that, to get more insights about the chemistry and also the biological annotation about that, we use what's so-called our tool called PROC chemotype plot, which is also in the Decoys website. So you can take it and can use it, and you can visualize some results. And you can really see here, you have the first bar that shows the chemistry information, the cluster, the rank coming from the docking, and the second bar that shows simply the bioactivity annotation. And what's really interesting here that you can really see from the docking rank 1 and 2, that means you have here active that shows the best ranking. So they are really enriched. You can really see it by the Fred, by the docking tool. And at the same time, they show the best performance, the best biological activity. Also, not only the best performance, but also the biological activity. And really sorry, you can really see from the red color they are also the most potent in a sub-micromolar range of activity, and that's yeah, this is what was very important guiding for us. And if you just visualize, you can really see they can simply show a superposition, optimal superposition with the modeled ligand. And you have more information like which clusters these actives are. You can also see to make some assessment of the scores distribution of each cluster, and also you can make some cheminformatics details like intercluster similarity, is there any bias, and etc. So this visualization was very important to us to get more information about the drug discovery and how to simply design more structures in the future. But during the reviewing of this article, actually a new X-ray structure for the small molecule, we did actually a homology model. They actually, and there was announced an experimental X-ray structure, and it was perfectly the same as we predicted. And that was very important for us, because during the reviewing process, we could simply also provide a solid validation coming from the experiments right away. And that was, yeah, that was like a direct validation of our approach. At the end here for this particular example, we simply go from benchmarking to recommend a certain docking tool, for example, like FRED. Another examples we did, actually, we worked, for example, it was my pleasure to work with Professor Hazazi for a master student, Layla Al-Ghunaimi, at the American University in Cairo. And actually, Layla simply did a good work. She simply also trying to reinforce drug discovery against the COVID to explore new binding sites. So this actually was the idea. So it's not only recommending a docking tool, like classically we do with benchmarking, but also to explore a new binding site. And this actually done to, we were actually the first to say, okay, there could be a palm allosteric binding site for the polymerase of the SARS-CoV-2. And how we did this, simply we relied on the similarity with the HCV, hepatitis C virus, because there are also palm binders, palm inhibitors reported in literature. So we collected that and we performed our benchmarking against the HCV polymerase. And based on that, because we have a very high similarity and identity sequence of both polymerases, we say, okay, now docking tools tend to have a similar performance on similar binding sites. This is a known actually fact by structural bioinformatics. And this is why we use it and recommend a certain workflow, a certain prospective virtual screening for this new binding site palm subdomain. Another example is also a collaborative project for a master's student in Zuwil City with Professor Rima Arafa, actually. In this case, not only to provide a new binding site or even to just recommend a docking tool, but also to select a certain conformation of the protein. So because we selected a protein which is called fascine. Fascine actually is a protein for cancer. And to make a long story short here, we clustered all the conformations available. And then we selected certain conformation that shows a good benchmarking results. And then we made further MDE simulation to get more insights. And then at the end, we also did the same. We recommended here a protocol for fascine drug discovery for the future candidates. So now, to have some take-home messages. So the DCOIS 2.0 as a benchmark sets, it's basically important to evaluate docking tools in structure-based virtual screening. Also, it's quite important to develop and evaluate artificial intelligence scoring functions, particularly in machine learning. Also, it's important to identify new and unvalidated binding site, as we did with Layla Al-Ghunaimi did actually. And also to identify certain druggable conformation of protein like Heba did in her master. So at the end, I would like to thank all of you for the audience and your patience, and also would like to acknowledge all the collaboration partners, especially at Tübingen University, Professor Frank Buchler, Dr. Simon Vogel, Matthias Bauer, also Professor Hassan Azazi from the American University in Cairo with the master student Layla Al-Ghunaimi, also Muhammad Ismail from the British University in Egypt, Professor Rima Arafa from Zuwil City, and also would like to thank Alexandria University, Kafr Sheikh University, Bibliotheca Alexandrina, High Performance Clusters. And yeah, that's all. If you have any questions, you're welcome. I hope you're not sleepy after this lecture. Thank you very much. Yes. Thank you. Thank you, Professor Aziz.
Thank you very much, Mr. Hassan, for the invitation. Dr. Tamer, can you stop sharing? Sorry. Thank you very much. Can you see my slides and can you hear me well? Very good. Thank you very much. So, as Dr. Hassan mentioned, this will be my title. I hope I'll have time for all of this. Sorry, I didn't realize that I had to speak only for 20 minutes, but we'll see. Okay. So what the challenge in the basin said, always I would like to start with this slide because there is a lot of confusion in literature about the base sensor and the base sensor. The first challenge is can the base sensor provide acceptable detection limit to meet the infectious doses or what is really needed by the regulating agency? What I mean by this, you can be infected by one bacteria, and the best sensor in the market can't detect less than 1,000, basically, say a few per mil. So they're really far from what is needed. Okay, can the base sensor really work with such real samples like what you see here in the right-hand side? In case of these biomedical, blood, urine, mucus samples, food samples, or cyanobacteria, green milky samples. Most of the time, if you expose your base sensor to these, basically you'll get what is called false positive result, and this result from the non-specific adsorption. Most of the time in the base sensor, you need washing liquids and washing steps. You start normally with the buffer, introduce your sample, and then you wash with the buffer. And the difference between the two is basically how much you have basically with the concentration of the analyte. Imagine that you want to do this in the field. So you have to carry these liquids around, and this will basically, you need extra time for this. The analysis time, you need the shortest you can. When you talk about rapid, you need less than 5 minutes. Is the base sensor specific and to provide reliable and quantitative information? There's a big question there. The cost is very important, especially if you want to use in biomedical because they don't pay really much. And in terms of recognition receptors, it has to have a long shelf lifetime. And something else, you need to implement the requirement for the regulating agency when you develop your base sensors. For example, there's specific detection limits, specific volumes. In case, for example, of the environmental samples. If you want to run environmental samples in the lab using the base sensors, you need to run minimum 100 ml sample. Without this, they will not basically accept your data. So you need to think about this carefully and develop micro and nano sensor, how you will analyze this. And also after you develop the base sensor, you need to adapt the base sensor to the current practice and setting with minor changes. You can't come to the hospital and tell them basically, my base sensor is great, I just need you to change your way of working. They will not accept this. So you need to think about this in advance when you develop the base sensors. Just if you remember the COVID time, it was a bad time. I think most of you did this sample collection, use this swab. And then after that, basically they elute the sample collected in buffer. Then after that, you can run basically the immunoassays, or you can run the amplification techniques like PCR, ATM amplification, and so on. We'll not talk about disadvantage for each one of them, but we'll talk about the advantage for each one of them. What we like here, the sample collection. What we like here in the immunoassay, the simplicity and the low cost. And what we like about the amplification technique is the sensitivity and low detection limit. So the question is, can we integrate the three of them in one platform? That's the question. And can we eliminate this step? Okay, that's what we're going to answer in our development in the next 5 minutes or so. So first, I'll start with really something simple. I don't want to talk about electrochemical sensors, so I decided to change my topic actually, talk about something a little simpler than this one here. So we're going to talk about cotton swab. The cotton swab you are using for cleaning your ears in the morning after the shower. So when this Saleh started his PhD with me when I was at Cranfield University in 2014, he's interested in basically in detecting pathogens in food and predatory processing plants like stainless steel, like the glass and the meat itself. So we looked for these 4 pathogens: E. coli, Salmonella, Staph aureus, and Campylobacter jejuni at the beginning. So the idea was really, really simple. Is basically you get the sterilized cotton swab, you oxidize it to have aldehyde group, so you can mobilize your recognition receptor, which you need to bind to the pathogen you're interested in. So you can use antibodies, aptamers, peptide, bacteriophage, phage display, whatever you name it, you can. And this depends on the specificity you're interested in. Are you interested in specific strain, or you want a general recognition receptor combined to all bacteria or viruses? After you mobilize this, you go back here, and you swab the surface you are interested in. Then what will happen? If the pathogens exist, it's going to be captured by this recognition molecule. Now, how you can see the bacteria? That's the biggest question at that time. So we came with this idea. We have these colored nanobeads, and each one of them, they have a specific color and have a specific recognition receptor for specific pathogen. For example, if I have the black, this one here, the black beads, it has basically a specific recognition receptor. We started with monoclonal antibody. So if you have this monoclonal antibody specific for E. coli, what will happen? And you dip this one here in the developing solution, you'll see that these basically the bacteria get sandwiched between these 2 recognition elements with the black beads. As a result of this, what you will see, the cotton swab will become black. If you, for example, you target Salmonella, you'll see the blue and so on. So we have different colors for different pathogen, which is nice. What you can see here is just summarizing a lot of work, almost two years work. You see here, we artificially contaminated chicken, glass, and stainless steel in the top with serial dilutions of these 4 pathogens. And basically, we did the same assay I mentioned before. What you can realize here, each one of these cotton swab for specific pathogen, they have different colors. For example, Salmonella typhimurium black, Salmonella blue, Staph aureus orange, and Campylobacter jejuni green. So we can basically identify the pathogen. What you can see here, if you look here in the serial dilution, the intensity of the color increasing as the concentration increasing of the bacteria in almost 4 of them. And if you look for this, to do this, you need just only 5 minutes. And each one of these basically assay we develop, we run PCR for it, for different concentration, like what you see here. So this assay 5 minutes as screening in comparison with 4 hours for the PCR. Now, can we get the detection limit, which is the PCR can get? Yes, but this depends on the recognition element you are using. Some of them are really good, some of them are really bad. So the key here is the recognition elements you are using. So to continue, then we looked for the specificity for this test. For example, this sensor developed for some Salmonella typhimurium. When exposed to other bacteria, almost there is no colors. The same thing for Salmonella enteritis, and the same for Staph aureus, and so on. Mahabbat, that Saleh, he finished his PhD a few years back. Mahabbat just finished her PhD a couple of months ago. We started actually all of this work before the COVID. Okay, so the cotton swab idea is before the COVID. So Mahabbat looked for the flu A and the flu B, serial dilution with different colors, just you can identify what the flu. And then we wrote a code in the mobile, just basically to detect the intensity of the color, and we can have a semi-calibration curve. So the whole work here is not for real or accurate quantitative. It's just to know roughly the virus load or the quantitative analysis we are interested in. And here in the right-hand side, what you will see, for this flu A sensor, when you test it or your cluster acted with MERS-CoV, HCoV, and the flu B, almost there is no binding. The same thing for basically the flu B. Also, we tested this with the MERS-CoV. This actually, the MERS-CoV is just a coronavirus which hit Saudi Arabia and Korea just a few years before the COVID. But we are lucky this virus didn't spread as the COVID did, because the mortality rate for it is very high, almost close to 35%. So what you can see here, this is the serial dilution of the virus, almost from 10 up to 10 to the power 8. And you can see the intensity of the green color, the intensity increasing as you increase the concentration of the virus load. What you can see here, these are real patient samples. So we tested 12 patient samples here, and we compare with the PCR, which is run basically in the hospital. So what you can see here in the PCR, for example, we can see the signals comparable with the virus load or the intensity of the color you can see in the cotton swabs. So this is very intense. And some of these you can see they're basically, it's very faint. So basically that's proportional to the PCR, which this indicates there's a good correlation between the two, but the timing is a lot shorter for screening. We did also the same thing for the COVID-19. That's basically logic development. What we are doing now, we're trying to do multiplexing. So if you remember in the previous slide, basically we used one cotton swab for each virus or each pathogen. The idea was we're asking ourselves, can we integrate different recognition elements in the same cotton swab? So for example, can we integrate 4 of these basically viruses assays in the same cotton swab? And that's what we are working on. So we did really good progress in this. And I believe, yes, we can do it, but I would like to delay this result hopefully for next year. Happened during her PhD as well. She adopted the same technology for MRSA, and she compared actually in her work the antibodies for MRSA and also the aptamers in this assay for the MRSA. And she found out basically the aptamer is a lot better than the antibodies in terms of specificity and the specificity. It's a serial dilution, and this basically some cross-reactivity studies with this one here. Also, we work with the hospital next door, King Faisal Specialist Hospital, with Dr. Fatma Hamlan. She's interested in adenovirus, basically. So the idea here, normally they will get infected in their eyes, but you don't know if it's bacteria or viruses, and that's result from different, basically, infectious agents. So we are targeting this study as a proof of concept at the beginning, the adenovirus, if we can do it or not. So we get basically the agent we are using here from a local company, which they are basically developing lateral flow for adenovirus. So they provide us with the antibodies, and they provide also with the gold nanoparticles, not the polymeric nanobead, which I talked about before. And these gold nanoparticles conjugated with adenovirus. So we did the same assay for the adenovirus, and what you can see here, this is a result of the serial dilution of the adenovirus. And then we infected this mice, and every day we're taking a swab, and you can see after 1 week, the virus load, there's a color there. After 3 weeks, the intensity of the color is really high. This means the virus load is really high. So now what we are looking for to collaborate with the same group, they will treat basically that mice. And what we should see, basically, if we swab every day, at the beginning, this will be very intense, and then it will be cleared out. So we can monitor basically the progress of the treatment. That would be nice if we can do. We adopted the same technology also with the same Dr. Fatma Hamlan, looking for HPV. Most of the people doing pap smear here above 40. And basically, they are using these tools for collecting the sample. And then after that, they send it to central lab to do amplification technique. And the most commonly one is using the PCR. So we used here in this assay, after collecting the sample, we use blue nanobeads, and we have antibody for HPV, which is provided by Dr. Hamlan. She uses it basically in her lab on daily basis. So what you can see here, after you dip these tools in the blue nanobeads, if there's a blue color here, this indicates basically the female infected with HPV. If it's clear like this one here, this indicates there is no HPV infection. All of these assays is just basically less than or close to 5 minutes, just as screening. So we are not saying that's going to replace the PCR. These are just screening. Instead of running PCR tests for everything, so at least do a screening and then you run, so you can limit the number of the PCR because the PCR are a lot more expensive than these assays. Now we'll shift a little bit for electrochemical. So if you remember, we talked about using these swaps for colorimetric assays. Colorimetric assays is nice, but what about if the person suffering from color blindness? So they will not be able to see the color change there and will not be able to provide us with accurate quantitative information. So what we did, instead of using these tools here, the plastic, we fabricated electrodes here and we used a cotton swab around it. And basically we did the same assay, and these electrodes can be connected to your mobile and you can get the data in your mobile. So Dr. Shaima Isa, when she was basically working in my lab at that time, she fabricated the sensors, did a lot of characterization, assay development, and so on. It's very, very simple. So we use here a competitive assay. So she adopted this to the MERS-CoV, and this is serial dilution for the MERS-CoV, and this is a calibration curve. And that's you see the calibration curve going down because we do a competition assay. And also we did this for the COVID-19, and this is a calibration curve for it. And it worked very nicely as well, and we were able to detect very low concentration and more accurate than basically the colorimetric assay. And this is the cross-reactivity study, this for the MERS-CoV. You can see the drop in the human corona and the flu A, there is no drop, and for the COVID-19, the same thing. Now what we are working on, instead of using just only one sensor, and these sensors are just basically printed on papers. So now we are basically have 8 electrodes here. So we can integrate a number of recognition receptors, and basically we can use the same assay for detecting the respiratory viruses in a multiplexed basically fashion than doing it for single analyte. Now we'll shift a little bit towards optical methods. And during the COVID, we get some funding, and basically the mandate was don't develop any readers, any devices. So we need to use what is really used in the market to basically to shorten the time of the development. And we're looking for some of the viruses. Al-Khumra virus, this virus actually is discovered in Saudi Arabia in an area called Al-Khamra near Jeddah, where basically when they import all the animals, they keep it for 1 month there until they do screening before they release them to the market. So we have to be assay for this, and they have interest in Rift Valley and the COVID-19. So here in this case, we're going to use the fluorescence assays, but we will not develop any reader. And the nice thing about this, you can get these fluorometers in single channel, like your mobile size, or you can get up to 8 of them as well. Or if you want to run large samples, you can use a microtiter plate as a high throughput screening platform if you want to. So that's one, I think that's starting. So we have at least a reader there just to start with. So during this work, what we were looking for, the proteases which are released by the viruses, not the virus itself. And we'll talk about this in more details later. So what we have here a random library of dipeptide, very short, basically, peptide sequences to eliminate the cross, basically, reactivity with other viruses. So you have a random library here, and each one of them has a fluorescence sequencer. And basically, you add a supernatant of the virus to each one of them and see which one can cleave and what you measure here, decrease in the fluorescence. So what you can see here is some result for the Khumra. We tested, I think, 115 peptide sequences for Khumra. And you can see some of these give us high fluorescence signal. So these are possible. We can't confirm these can be used specifically for Al-Khumra virus. These are possible to be used as hits for Al-Khumra detection. So we get these ones here, and basically we just normalize the data here, the fluorescence for these sequences, and we do a lot of cross-reactivity of the sequence we selected with other viruses to see which one cross-react. And the one which didn't cross-react, then we can use it as possible substrate for detection. Here are some of the results. So we selected one sequence, which is basically can be cleaved only by Al-Khumra virus. And we incubated it with serial dilution of the virus from 10, almost 10 to the power 8. And we monitored the increase in the fluorescence up to 140 minutes. What you can see here, you look here in the x-axis, around 20 minutes here, what you can realize is we can get more than 60 to 70% of the signal. So we don't need to run for that long to see the cleavage. So after 20 minutes, if you plot the fluorescence after 20 minutes here, as a function of the virus concentration, you can see it give us a linear correlation. And that's what leads us basically to the calibration curve here. And you can here see the limit detection for this is 15.5 PFU per mil, which is really, really nice. And some of the application even it's better than the PCR as screening method. So you don't need, because this assay is a lot cheaper than using basically the amplification. I mean specifically is the PCR method for screening. Then for confirmation, you can go and run the PCR, but it's a lot more expensive. We did the same thing for Rift Valley, possible hit, and these are the hit in comparison with other peptides. And basically, we did the same thing, incubating this with serial dilution, and we constructed the calibration curve. And surprisingly, it gave us really good detection limit too. And this is some of the cross-reactivity with other viruses. We did the same thing for the COVID. Sorry, we hid all these in here, the peptide sequences, because we still didn't publish this at all. So these are new data. For the COVID-19, yes, we published this a couple of years ago, but for the other virus we didn't. This is basically the cross-reactivity. And then after that, we collaborated with the hospital, I think with also KAU from Jeddah University, King Abdulaziz University, and we ran the FRET method with the PCR, and they gave us a very good correlation. The advantage of this technology, all you need just to have a peptide sequence substrate with the fluorescence sequencer there, and after you collect the sample, just dilute it there, and then incubate it for 15, 20 minutes, and you can run this in the field or basically in a lab environment. As you saw from the detection limit, it's really highly sensitive. It's very simple assay. I don't need polymerase, I don't need primers, I don't need anything else. No need for sample processing like what you do in the PCR, virus slicing and extraction and so on. And all you just need a peptide sequence, which is highly robust, unlike basically if you want to use the polymerase and so on. And for the cost, it's less than 10% of the PCR cost. So for screening purposes, not for confirmation, for screening purposes, I think this is a very good assay to be used instead of basically the PCR. And this can be run in the field or in a high throughput screening in lab settings. And the advantage of this, you can have a multiple substrate for doing multiplexing. So you can have a substrate for every virus or every pathogen. And basically you just change the fluorescence sequencer for each one of them. And you can do multiplexing with this basically technology. The nice thing about this now, we developed in homogeneous assay, but also you can do it in a paper-based assay. And that's what actually it led us to this work, to develop a paper-based sensors. And this is really simple. This Sahar started when I started her PhD with me 6 years ago. This is the paper assay. So we have the peptide sequence here, very short peptide, ditripeptide maximum. And we add a spacer here, and we add a spacer from the other side. And we conjugated the top of this spacer to black magnetic beads. When we attach this to the paper here, what you will see, the paper become black. So what you see here, this is a black line here. This is from the attachment of the magnetic nanobead, the black magnetic nanobeads conjugated with the peptide with the spacer. Forget about this color. These colors indicate nothing because that's we started on a pH paper actually. So and we did this on a pH paper. So these colors mean nothing. Okay. So now we have this black color. So when you add the protease there, what will happen? The protease should cleave this one here. So, but after that, what will happen? The black beads will still sit on the sensor itself. This basically, I have to incorporate washing steps. in during this assay, but we're trying to minimize this. So what we did, this is the top surface of the paper. The back of the paper here, what you can see here, there's a black here magnetic paper. This actually, it's the magnetic paper you basically stick in your fridge. We buy 1 meter by 1 meter with less than $5. So you cut them to small pieces and attach them in the back of the paper. Why we did this? The moment basically the protease come and cleave this, what will happen? These black beads here will be dragged in the magnet here on the top of the magnet paper. So I don't need a washing steps and we can eliminate this completely. And that's what you'll see here. The black, after you add the protease, what will happen? You see the appearance of the color, of the yellow color, the base of the paper, and also here that basically that dragged basically magnetic beads on the magnet paper. When we started this work, we started using basically paper cutter, as you see here, and doing single sensor, or we can do multiplexing, limited multiplexing number up to 3 here. And now we also automated this to fabricate 50,000 sensors per day. And I think the last 5-6 years, we published more than 20 articles about this technology. So Sahar, during her work, she basically artificially contaminated these food matrices. And this is before and this is after. And you can see every time there's the yellow color appeared here, and also the accumulation of the magnetic beads on the magnetic paper. That's indicate basically there is a bacteria, bacteria in this case, because that's what they artificially contaminated. This for the staph, and this the result here, the E. coli. Also, Sahar worked with Dr. Ghadir. She was a visitor. She has actually used to visit my lab every year from the University of Jordan. They adopted the same technology for basically Listeria. This is before and this is after. And also by using the phone, you can write the code to have semi-quantitative, and we compare the D and L amino acids and so on. And this is the crystal activity, and thus this is some real samples. We adopted this technology. Yes. All right, okay. We adapted this for mastitis and for also botulinum neurotoxin as well. There's another work for another PhD student. Also for the B. gingivalis as well, which is called peritonitis. You can see here, this is serial dilution. And this actually patient samples. We tested more than 420 patient samples, patient with basically documented B. gingivalis for peritonitis diseases. And some articulators ask us basically to integrate H&E and cathepsin G as basically as proteases in this paper. We did single sensor like what you see here. And these are basically double sensors, 2 sensors, or dual sensors, when exposed to different concentration of the proteases. And this is actually patient samples. We adopted the same technology for Pseudomonas aeruginosa, and these are serial dilution. And this basically is a patient sample from King Faisal Specialist Hospital. We did also anthrax as well. And Dr. Ghadir, during last summer, she came to my lab and she did also the black mold. And we applied the same technology for Legionella detection. And we run this for 6 strains of bacteria, what you see here. And what surprisingly we find, the 5 in the blue, they can basically cleave 1 peptide sequence. So you can use 1 sensor for detecting the 5 of them. And the purple color, so we need to integrate single sensor. We adopted the same thing for Brucella and Selmatufi. And the nice thing about these sensors, just on my last slide, is we can print them in paper, in sellotape, in a cloth, and so on. So we integrate them in many applications, like one of them here. Basically, the container of the contact lens. When you put the contact lens, if you wake up in the morning and you find it change the color, don't wear this lens because it's contaminated. And also integrate this for this dialysis bags. Not dialysis. The peritoneal dialysis bag basically here. In the pipeline, we put these guys to disconnect a number of times here. So in the drain, we integrate these sensors here, and you can see when I have basically infection, the color of the sensor change. So we did more than 70 applications of these applications. So I'll stop here, and I'll be happy to take any questions. I'd like also to thank you for basically your attention. Dr. Hassan. Yes, I'll be happy to take a question. Yes. Yes. We tried actually both. We tried both actually, and both is working. But as you mentioned, one of the biggest question is the reproducibility. But since you're using for screening and you don't want to do quantum information, I think it will work. Both will work. Yes. Thank you very much. Inshallah. Inshallah. Thank you very much. Thank you. Nice to talk to you. Thank you. Take care.
ابتدي. اوكي اوكي. تمام يا فندم. تمام يا فندم. تمام. Chairman, Mr. Chairman, my colleagues, everybody, good afternoon. It's my honor and pleasure. It's my honor and pleasure to be invited to such an important event, and I thank the Professor Azazi and the organizers for letting me share with you some highlights about the field of ancient DNA analysis and paleogenomics. Paleo stands, as you know, for something ancient or old. Is it okay, everybody? The voice is okay, sound is okay, and okay. Okay, thank you very much, Professor Hassan. Okay. Let me start by saying what's a mummy, because it's a little bit different from what you call biological remain. A mummy is human or animal remains with preservation of non-bony tissue. It has flesh or even flesh and wrappings, which is different a little bit from having skeletons in as a biological remain to study. Usually the soft tissue are preserved when the post-mortem conditions favor dehydration. And it can be natural if somebody dies and the body is kept in very dry and hot weather, like in the desert, or in a very cold climate, very dry and cold climate, like, for example, in the Alps, or high in a very snowy environment and cold environment. On the other hand, artificial mummification, a science which has been excelled and peaked in its art by ancient Egyptians, or what we call embalming or mummification. It's aimed to preserve the body, and because of the religious belief that it will guide the soul back to the body and improve the person's chance of passing to the next world. The ancient DNA field is relatively a young field. It has only started in the mid-80s by the very first paper in the English literature in 84 by Higuchi, following by another paper by Savanti Pabu in 85 about an Egyptian mummy. And just to remind you, Savanti Pabu got already a Nobel Prize winner a few months ago in medicine for his pioneering work on the Neanderthal and the human ancestors. In the 90s, the field took off with the discovery of PCR, because actually, usually the ancient DNA is in a very bad state. It's very low copy number and highly degraded. So actually, the PCR came in its rescue for that, and it revolutionized the field. But the other side of the coin, that it has also increased the possibility of contamination and generating irreproducible results. That's why about a set of criteria or guidelines for ancient DNA research, particularly if you are dealing with human remains, that the scientific community embrace, so they can, what we call, authenticate the result that's coming out. And the first thing is, the ancient DNA lab has to be a dedicated lab. It's a lab that doesn't allow modern at all. And fortunately to say that in Egypt, we have 2 labs, and actually that's another one of the guidelines, that there has to be 2 labs for replication. The one that's present in the basement of the Egyptian Museum, and the second one, it's present in the National Museum of Egyptian Civilization. Each lab, and these are probably apart from ones in South Africa, are the only ones in Africa, and apart from labs in Israel, are the only ones present in the Middle East too. So they are unique labs. They are the engineering design that they are composed of physically isolated areas, with particularly the extraction room has to be isolated. There is one-way movement in the lab. It has positive displacement air conditioning. It has UV lights to sterilize the air that's turned on each night. We usually, we always have protective clothing when we go into the sterile area. There is strict contamination removal. The flooring is smooth, no cracks in it. The walls are painted with antibacterial, and there is always also a laboratory records. The molecular behavior are closely monitored. So if you get PCR fragments that's exceeding 200 base pair, that's questionable that it's coming from ancient, because the major fear that the sample is contaminated by modern DNA, because usually the enzyme system for the PCR and so prefer the intact DNA rather than the highly degraded DNA from ancient tissues. So if you get a PCR fragment of 500, this is exactly strictly not ancient DNA at all. It's mostly contamination with molecular with modern DNA. Of course, you will do negative control in each experiment. There has to be reproducibility of the result. That's why we have 2 labs with 2 teams doing the same work. And we also know that the genotypes of the lab staff. So if we get a result that's kind of similar to the DNA fingerprint of the lab staff, it means that it's contamination from the lab staff. And usually we take samples. I usually prefer to take samples using as much as possible aseptic techniques, using a bone marrow biopsy needle to minimize the damage to the human remains, particularly when we are dealing with such precious subjects like the royal mummies. And we can have some guidance from the CT scan to go into the areas where it's mostly covered with flesh and with wrappings if possible, and to get this kind of strictly aseptic samples from the inside of the mummy. And this is the sampling in this. This is the sampling of Tutankhamun it's done also aseptically in the tomb. So it's not only we follow this in the lab, also in the tomb. This is the tomb of Tutankhamun KV 62. And you see the amount of tissue that we take. It's as pinhead. It's quite small. So sorry. So why we do this? Why we do ancient Egyptian genomics? What's for? Let me speak about the methodological approach first. DNA extraction with stress on authentication and contamination control, as I previously elaborated on. Then we go into optimization of ancient DNA extraction method for each sample, because actually it's quite challenging to work on ancient DNA, because actually each sample has its own case or merit, whether because this depends also on the resin that has been used in mummification, and there are different components and different cocktails of resin over the years. It's a science that has evolved over thousands of years with the ancient Egyptians, and in this way, not all the samples are the same, not all the resin components are the same, and its effect on inhibiting the reaction or affecting the DNA retrieval differs from one sample to the other, even within the same body of the same individual. Because actually, one of the intriguing things that was discovered by our colleagues in National Research Center and Tübingen, that for mummification, they use more than one resin cocktail for the same body. Some cocktails for the head, some cocktail for the trunk. So actually, it's very challenging to get DNA out of this. So we have to optimize this and adapt this to each sample that we are working on. Then the standard now, which is doing the standard platform, as I will elaborate later on, we're using NGS, next generation sequencing. We may use some target sequencing or target analysis if we are looking for a certain area, or using enrichment, which is to increase the amount of certain DNA sequence, like mitochondrial DNA, for example, or certain genes that we wish to study. So there are two ways to do this, either to do targeted analysis for a certain area of the genome, or metagenomic analysis, where we will collect all the DNA within the sample. Then we do the dry lab. نعم يا فندم. حاضر يا فندم. Then we do the dry lab analysis of the generated data. This is one example that was done about 10 years ago, our study on the 18th dynasty, to draw the family pedigree, where we identified KB35 elder lady as Queen T, and she is shown already in the NMEC now. The young lady is the mother of Tut, and the KB55 Akhnata. And we could identify that malarial sequences in 4 subjects, Tut and 2 of his grandparents, Tuya and Yuya. The next generation sequencing, as I said, is a standard platform now, and the first publication that came out on next generation sequencing, ancient human, was coming out in 2010. And the first one coming out on Egyptian mummies was coming from my colleague and ex-student, Khairat, Rabab Khairat, during her PhD thesis in Tübingen. And she showed that the next generation sequencing data showed there is deterioration of the studied mummies, and it correlated with the increase in the microbial load, the microbes that eat out the flesh of the mummy. So the applications of next generation sequencing, it will help in studying of the ancient human genomic data for diagnosis of rare diseases or conferring susceptibility to disease, comparative analysis of the ancient microbial genomics with the contemporary ones to reveal the evolutionary history of the microbes, establishment of familial relationship of humans and shedding light on their migratory movements, studying the content and amount of microbial load in the archaeological specimen, guiding its preservation and conservation plans. And lastly, we can analyze the DNA, the collective DNA that we gather in the metagenomic sample for animals, humans, or plants, and classify it in its taxonomic family and to detect if we have any infectious, if they have any infectious disease during life. These are the collaborators and the funding agencies that we are dealing with. Thank you very much for your listening.
Okay, thank you so much for the invitation and introduction. Let me first share my screen. Can you see my presentation? Okay, perfect. Thank you so much. So today I'll be talking about genetic risks of Parkinson's disease in Egypt. And throughout the presentation, I will try to answer three questions. Why would it be appealing to study neurodegenerative disease? Why it is important to study genetics of such diseases in underrepresented populations like in Africa in general or in Egypt specifically? And finally, I will highlight some of the recent findings from our research done on the area of genetics of Parkinson's disease in Egypt. As you can see, the work is funded mutually by the American University in Cairo, the Global Parkinson Genetic Program, the IPDGC Africa, and in collaboration with the University College London and the Egyptian Network for Neurodegenerative Disease. So the first question is, why should we start working on neurodegenerative disease or understanding the risk factors for neurodegenerative disease? And the answer, as you can see, because the prevalence of such diseases, whether we are talking about dementia, Alzheimer, or Parkinson's disease, is overshooting. And the tricky thing is that the rapid increase is more pronounced in the low and mid-income countries. However, most of the genetic studies or major studies concerned with identification of risk factors are coming from developed countries. And maybe actually this is the reason why the prevalence is decreasing in those countries and increasing or overshooting in underrepresented population like in Africa and Egypt. Second, why should we dig deeper and try to study genetics of neurodegenerative disease in general, or any type of disorders in Africa? As you can see, because first, based on most of scientific research, migrations are coming out of Africa, which means that African genome would be rich source for better understanding or the identification of risk factors due to the fact of lower linkage disequilibrium, more haplotype blocks, high degree of within population admixture. In principle, we can say that the African genome is more diverse and more rich. This is a recent publication showing that the African genome size is actually bigger than the human reference genome based on European ancestry. And this figure also shows that including genetic data from underrepresented population could help to better understand and identify risks or develop even polygenic risk scores for global knowledge and for global understanding of the reasons of the disease. So including genetic data from Africa, Middle East, and Latin America, and different underrepresented population is mandatory to better understand and characterize risk factors for disease. Ironically enough, however, is on the contrary to the previous data shared with you, you can find that most of genome-wide association studies published to date, the majority are coming from Europe or North America. As we can see, Africa is only contributing by 2% of all GWAS or genome-wide studies worldwide, and we need to change this. This is a recent paper published a couple of years ago showing that the Egyptian genome is somehow different from the well-studied genomes worldwide, and this necessitates digging deeper and specifically studying genetic risk factors in our population as an example of understudied population. When we dig deeper and when we focus on the genetics of Parkinson's disease, we find out that prevalence of Parkinson's disease in Egypt, most of the studies are localized, not nationally representative, but at least they can enlighten us that the prevalence of Parkinson's disease in Egypt is much higher compared to international standards. This means that risk factors might be higher. This means that genetics of the disease could be different. Still, is an understudied area and understudied population. And this was the reason why we started or initiated the Egyptian Network for Neurodegenerative Disease maybe 10 years ago. And as you can see, the network is represented in most of Egyptian universities and Egyptian cities. It's a collaboration between neurologists, neuroscientists, geneticists from different places, trying to unravel the reasons of neurodegenerative disease and understand the high prevalence of such disease in the Egyptian population. Previous efforts for the END or the Egyptian Network for Neurodegenerative Disease led to some publications. However, currently we are conducting what I consider one of the biggest projects in the area of Parkinson's disease through establishing the first Egyptian cohort, which is geographically distributed among Egypt, representing different places in Egypt. And we have 1,000 subjects. Those subjects will be enrolled for whole exome sequencing alongside single genetic analysis or targeted sequencing for known pathogenic variants for Parkinson's disease. As you can see, this is the structure of the cohort recruited so far. And as you can see as well, it is distributed across different cities or governorates in Egypt. And this is also this section of the distribution of cases and the phenotype, whether they are controls or Parkinson's disease, and also a section regarding their age of onset. And in Parkinson's disease, the age of onset is sometimes correlated to the genetic risk factors or the type or makeup of the disease. So most of the time, early onset disease are considered, or early onset Parkinson's disease is considered genetic, mainly familiar form of Parkinson's disease, and late onset is mostly as the sporadic or idiopathic form. However, in Egypt, we find out that this data is not accurate or cannot be reflected on Egyptian population, because most of the patients recruited, even in sporadic cases, are early onset, which is a remarkable finding, by the way, even before conducting genetic study. And also, this is a dissection of the cohort according to consanguinity or history of family disease, family disorders. However, through our research, we succeeded to identify certain or study the condition of certain known risk genes for Parkinson's disease in our Egyptian population. The first genes that we were exploring is the LRRK2 group. And the value of LRRK2 is actually that LRRK2 is the most important, most prevalent gene variant for Parkinson's disease. As you can see, we have now world map of reported LRRK2 mutation. The value of LRRK2 also is that now it is the first targeted therapy for Parkinson's disease. So when we're talking about precision health or personalized medicine, clinical trials now are running to trying to identify, and actually they have identified some protocols for treatment of specific type of patients, which we call LRRK2 Parkinson's disease. And that's why they are trying now to establish a world map. So in population where LRRK2 is prevalent, it's very appealing to conduct such clinical trials. It's very appealing and interesting to include those populations in the studies. Definitely, the highest prevalence of LRRK2 mutation is found in North Africa, specifically in Tunisia. They have very well characterized cohort with perfect clinical history as well as genetic testing. However, in Egypt, we didn't have the chance to conduct a robust or solid analysis. Few distributed research was conducted, and it was always localized. So one of the research was done in Alexandria, and the other one was in Upper Egypt. And as you can see, the prevalence is contrasting. So in one of the studies, the prevalence showed around 10%, and on the other side, it's less than 2%. And it is challenging actually to build policies or plans based on those contradictory data. However, our findings show that the incidence or the prevalence of LRRK2 in Egyptian population based on a big cohort like ours is around 5%, which is a good number. Which is a good percentage. So when we're talking about Tunisia, which is the highest prevalence worldwide, we're talking about 30%. However, when we're talking about Europe or North America, the prevalence is very low. That's why we are in a good position. We are in a good situation. We are not as good as Tunisia, as I mentioned, but we are much better than other countries. So this makes Egypt eligible to be included in the LRRK2 club, as they say. So this makes Egypt appealing place to start thinking of clinical trials or thinking of adopting precision health solutions for Parkinson's disease tailored to LRRK2 mutation. This is the first study, and I would say the first study with robust data based on the number of the cohort and the diversity of patients and controls to prove that we have LRRK2 mutation prevalent, the most common type of LRRK2 mutation. And also, as you can see, we have homozygous and heterozygous, and we even have LRRK2 mutation in controls, which is very interesting. And this highlights the value of penetrance and the possibility of gene-gene interaction or even gene-environment interaction. And this is also the distribution. And the remarkable thing, as you can see, is that it is not localized in certain governorate. It is distributed. This means that LRRK2 mutations are widely distributed among the Egyptian population, and we have higher rate and high prevalence of LRRK2 compared to European population, meaning that we are more in comparison to Tunisia, even if we are not as high as Tunisian population. So the second variant or gene we try to explore is ApoE. ApoE is known variant or known risk gene for Alzheimer's disease. However, recently they started to discuss the possibility of playing a role in Parkinson's disease as well, given the overlap between different neurodegenerative disease. Unfortunately, I don't know if it is unfortunately or as usual, we couldn't find any previous genetic study for ApoE in Parkinson's disease in Egypt, maybe only one in Alzheimer's disease. So we tried in our cohort, and according to our results, ApoE cannot be correlated to Parkinson's disease. And by the way, this is aligned to most of the research worldwide. So the risk variants we identified are not correlated to pathogenicity of Parkinson's disease or by the risk of, or cannot be correlated to the risk of developing the disease. However, we need now to dig deeper into the phenotypes. So based on previous research, ApoE might be related to the cognitive decline, which is one of the features of Parkinson's disease. So usually we have either Parkinson's disease without dementia or without cognitive impairment, or Parkinson's disease with dementia, or what we call, in other words, Lewy body with dementia. So this is worth mentioning, worth further studies. Now we are in the stage of digging deeper and correlating the data to the cognitive functions of our patient to see whether ApoE, which is not now identified as a risk gene for Parkinson's disease, could be a risk gene for developing of cognitive impairment in Parkinson's patients. So this is another finding we identified from our cohort. As I mentioned, it's not a risk gene for Parkinson's disease so far. However, we need to know whether it can induce or promote or affect the cognitive ability of Parkinson's patients or not based on our MoCA and mini mental state tests that we have done to our patients. The third one is the MAPT, which is the gene for tau. And tau is one of the neuroskeletal proteins, and it has been involved in several neurodegenerative disease, given a term named tauopathy. And tauopathy is a spectrum of disease involving Parkinson's disease, Alzheimer's disease, Okay, so we evaluated the impact of tau mutation in our cohort, and we've proven that, at least in our findings, MAPT can play a role as a risk gene for Parkinson's disease as well, which is a remarkable and new finding compared to other studies. Finally, the K variant, which is a validation of our previous study. K variant is a gene encoding detoxification enzyme, and based on our results, it is not correlated directly to Parkinson's disease. However, if we added risk factors like pesticide exposure or toxic exposure, it can help or increase the vulnerability of developing a disease, which is also highlighting the value of studying gene-environment interaction when we are studying idiopathic diseases or multidisciplinary diseases like neurodegenerative disease. To sum up, we collected or recruited the largest cohort of Parkinson's disease in Egypt. We identified or at least characterized the prevalence of LARC2, which is the commonest pathogenic variant among Egyptians, with a prevalence of 5%, which is much higher than the estimated worldwide. Again, this enable Egypt to be part of ongoing clinical trials on studying the impact of personalized medicine on Parkinson's disease. ApoE genotypes are not responsible for Parkinson's disease, but we need to correlate to cognitive impairment. MAPT H1 seems to be a risk factor for Parkinson's disease in Egyptians, pending further investigation. Currently, we are trying also to study gene-gene interaction, like the role of MAPT and ApoE or ApoE with K variant, whether they potentiate actions or increase the penetrance of each other. This should be the next step in our study. Thank you, and thanks for all collaborators. Thanks.
Yes. Did you hear me? Okay. Okay, thank you very much. Okay, thank you very much. First, I would like to thank the organizers of Egyptian National Community of Biochemistry and Molecular Biology for this great effort. Bismillah ar-Rahman ar-Rahim. My talking is about the impact of the BRCA gene single nucleotide variants in glioblastoma multiforme among the Egyptian patients. Glial cells are mainly the astrocyte cells that can grow very quickly, causing the aggressive types of the cells called glioma. Glial cells is glue cells located in the neurons and involving the nutrition and maintenance of the nerve cells. So the cancers, brain cancer can divide it into two main types. Glioma, this is responsible for the massive growth of the astrocytes, and non-glioma cells. Glioma divided into 4 different stages.
دكتور مجدي ممكن حضرتك تكبر السلايد بعد إذنك. دوس على البتاع اللي على اليمين ده.
Okay. That's okay.
شكرا. تمام يا فندم.
So glioblastoma is divided into 4 different stages. Stage 1 to stage 4. The most aggressive and progressed stage is the stage 4 that's called glioblastoma multiforme. Glioblastoma multiforme is one of the deadest malignant tumor in the world, which account for about 32% of the central nervous system tumors. So we are concerning about these types of the brain tumor. The symptoms of the glioblastoma is common, like the nausea, vomiting, headache. This is common for many diseases. So we can diagnose the glioblastoma by 2 main techniques, CT scan and MRI scan. The clinical treatment of the glioblastoma is mainly by the surgery, removal of the tumor, followed by radiotherapy and chemotherapy. The main cause for the glioblastoma is defect in the function of one or more of the pathways in the cell. DNA repairing is the most significant pathway responsible for the glioblastoma according to the different studies. DNA repairing involving in 34 different genes, and the 55 effective SNPs are involving in this disease. So we are concerning about the repairing disease. BRCA genes are repairing genes are susceptible genes for the cancer. BRCA1 and BRCA2 are the 2 main types of the BRCA genes that prevent the abnormal proliferation of the cells, and they were studied in the breast and ovarian cancer, but not until now studied and documented for the GBM disease. So we try to find the relationship between the nucleotide variants in the BRCA genes in relation to the glioblastoma. BRCA1 are the genes located at chromosome 17, position 21, with 24 exons, and BRCA2 located at chromosome 13, at position 12, subtype 3, with the 26 exons. Each of them containing more than 36 different mutations. Our experiment designed starting with using selected 15 adult patients, 10 of them females and 5 males. They newly diagnosed with the GBM, aging about 55 years old. They don't receive any kind of treatment and don't suffer from any other types of the malignancy. And the scale of performance of the tumors according to the ECOG groups concerning the oncology were selected, in addition to the 5 controls, 2 of them with family history and 3 without a family history of the GBM. Refers to the treatment of the patients. In our experiments, 4 patients were totally excised of the tumors, and 11 patients were excised subtotally and with biopsy. Then followed by the treatment of the radiotherapy and the chemotherapy. 10 of them suffering from the radiotherapy and the chemotherapy with temozolomide with 6 months for 6 cycles. 3 patients treated with the radiotherapy and chemotherapy for 3 cycles, and 2 patients only treated with radiotherapy. In response of the treatment, 6 patients were partially response, 4 patients were completely response, 3 patients were progressive disease, and 2 patients with stable disease. So we are to identify why after this treatment, 3 patients with a progressive disease. So we concerned about the BD patients, why they not progressed after the treatment with the chemotherapy and radiotherapy in relation to the mutations of the BRCA genes. So using the ion torrent AmpliSec techniques for BRCA1 and BRCA2, using the ion torrent versions for the data analysis, using the NCBI reference genes for the BRCA1 and BRCA2, then the data were submitted to the link disequilibrium for measuring and analysis the haplotypes of each genes. We got about 31 genetic variants of the 2 genes, 13 of them from the BRCA1 and 18 from the BRCA2 mutations. All of the 13 BRCA1 mutations were previously published in the database. 10 of them are exonic and 3 are intronic. From the 18 single nucleotide variants of the BRCA2 genes, 15 of them were previously identified and reported in database single nucleotide variants, and we discovered 3 de novo variants among the Egyptian patients. 1 deletion in introns and 1 deletion in exons, in addition to 1 deletion in 1 single nucleotide mismatch in introns. So we have 3 new variants, 1 mutations in introns, a single nucleotide variants, and 2 deletions in exons. We try to find the pathogenicity of these genes according to the different types of the program. So we applied all the variants to different types of the programs like polyphen 2, SAFE, Panthers, mutationals, Profin, I mutant, to identify if these mutations are pathogenic or not. In total, we find that all the 13 detected genotypes of the BRCA1 are ranging from the benign to the likely benign pathogenic. Among the 18 detected single nucleotide variants in the BRCA2, 14 of them were likely to benign, while 1 is deleterious, and the 3 discovered new mutations among the Egyptian patients were recorded as a pathogenic variant as far as we know till now, because we don't have any references about the pathogenicity of these variants. This is the summarize of the mutations in the BRCA1 genes, 3 intronic and 10 exonic. We submit all these 3 variants to the hot HapMap types to identify the interrelationship between the different types of the single nucleotide variants in the BRCA1. So haplotypes detected that there are 8 BRCA1 single nucleotide variants are related to each other, is depending on the high allele frequency aggregators techniques, and 4 SNPs are weak related to each other, and 1 SNP with not related to any other of the nucleotide variants. This hot map refers to the interrelationship between these 13 different variants. The red colors means the interrelationship. The darker color means the high, strong relationship between each other. So we select from these maps about 8 different relationship haplotypes, that is getting the higher score for the haplotype counting, ranging from 1773 to 55. And then we try to minimize, to make more concentrations about the haplotypes among these mutations. So we select the most higher 5 haplotypes, more than 106. So we find there are 5 main haplotype mutations, related to the 13 patients in the BRCA1. We did the same things for the mutations in the BRCA2. So this is the red color refers to the new identified Egyptian mutation. This is the exonic mutations, and this is the intronic mutations. This also two new deletion mutations among the Egyptian patients, and we applied the same techniques for the HapMap type. So BRCA2 finds there are 6 only different mutations related to each others and can account for the high effect on the GBM disease. So okay, so finally we have 5 haplotypes A, B, C, D, and E. A, B, C, A, B, D were briefly discovered, and we divided 2 new haplotypes C and D. In relation to the disease, we find the patient number 2 have haplotype A, patient number 11 have haplotype AB, and patient 15 haplotype E. These 3 patients are progressive disease. So, and finally, A and new discovered B and C are related to the good response to the timidazole treatment for the GBM, while haplotype E may relate it to the poor response to the timidazole treatment and higher risk for the GBM. Haplotype B may be agreed. So if you apply this to the normal patients, so we find the 3 patients without family history are in the haplotype E. This is very consistent with our result, while the 2 patients with the haplotype A and B maybe has a high risk to the GBM. So in conclusions, it is first studying for the BRCA genes in the genomic patients referred to the GBM using the GBS. It is the first haplotyping referred to the BRCA genes using the samples from the Egyptian patients. The current study emphasizes the relationship between the BRCA gene mutation and GBM, which may be open the new patches for the treatment for the patient resistance to the idazolamides. And if the patients are resistant and this have the genotype E, we may in the beginning refer to the another chemotherapy treatment instead of saving time and patient's health. So further studies are needed for the large patients to control to need to investigate the different role of the BRCA1. Future perspective determines the mutations of the patient and their families, studies assumptions of the large scale, and looking for the new bioinformatic tools to confirm the haplotype assumption. This is the paper published, and thank you. Thank you very much.
Thank you, Dr. Muhammad, for this nice presentation. So I would like to ask, why don't you try to dig more for these mutations in some large databases like TCGA or something like that, to see if any BRCA mutations are there over there in this database?
We already did the assembly using the BLAST in the NCBI and found something between about 13 mutations was previously published in the TGA and identified the pathogenicity also of them. We are concerned about the new Egyptian mutation.
So the two new mutations you found was reported in this database in the TCGA?
No, not reported. This is new Egyptian mutations.
So it's inherent only in the Egyptian population, right?
Among the Egyptian population, right?
Thank you.
Okay,
Thank you, Dr. Hassan.
Welcome. Thank you, Dr. Hassan. You're welcome.
Dr. Azizi, thank you so much for the conference and for the great organization and for introducing me this way. I'm glad to share the current work which we are doing together in this conference. So my screen is clear right now, right? Okay, should I start? The screen is, can you see the screen, everyone?
Yes, it's clear.
Okay, so I'm going to talk about single cell analysis of healing versus non-healing diabetic foot ulcer. So this work actually I was doing with Shireen, the major part of this work, along with collaboration with different departments from the AUC, like biology, public health, computer science, and other collaborators from the US, like Indiana University and Pittsburgh University. So today I'm going to present this work. So I will give you a very brief introduction about the problem we are trying to solve. That the diabetes, of course, some of the patients can develop diabetic foot ulceration in their life, and this ulcer can affect them, affect their life significantly, like prolonged hospitalization and feeling like depression, and the most worst complication of it, like the amputations. And in the US alone, 70,000 permutation can happen per year. So along during the previous few years, I worked with Dr. Azizi and other collaborators in the US. So we published multiple papers to improve the wound healing in chronic wounded patients or in diabetic patients. And in one of them, we compared between healthy skin and diabetic skin, but without ulcer. And we identified some critical factors or components having defect in the diabetic skin that can affect the vasculature development in them. But this time we downloaded a new data set. I will talk about it in the next slide. For the ulcer microenvironment, because this is the problem, that the ulcer microenvironment itself and the healing versus non-healing with different cell types interacting together, it is very complicated. So the aim of this study, we want to dissect this microenvironment for each cell type alone to study the interaction between them and to identify what are the master regulators that can affect the global pattern we can observe in the microenvironment of this ulcer. And hopefully we aim to induce transition of the non-healing cell states towards the healing states across all the cell types simultaneously. The kind of data we are using, we are all familiar with. As we know that the DNA can be transcribed to form the messenger RNAs, and when we estimate the level of mRNAs in different conditions, we can compare between two conditions together, and higher abundance of mRNA probably can be translated to higher amount of protein. And this is the kind of data we are working with, like we are counting the mRNA, but at level of single cell. So we are estimating the RNA level at level of single cell from healthy skin, diabetic skin without ulcer, and diabetic ulceration, either healing group or non-healing group. So the data was retrieved from the GEO database using this accession number for now. We started with clustering the cells. We have around 100,000 cells, and after filtration and removing low quality cells, 86,000 cells remained, and those are the major clusters. And cells basically can be clustered together based on the gene expression profile for each cell. So similar cells or similar cell type or subtype can be clustered together, as we can see here. So the next step is to compare to identify what are the marker genes for each of those clusters. So we can obtain this by comparing the gene expression level in cluster 0, for example, versus all other cells, or cluster 1 versus all other cells. And from this step, we can identify the top markers or the top genes expressed in each cluster. And from those genes and comparing them with the markers from literature, we can identify the cell types. So basically, we have those 4 conditions, and we identified fibroblast keratinocytes and some other immune cells and endothelial cell or vascular endothelial cells as well. Next step, we focused on comparing the 2 conditions, diabetic skin with healing ulcer or with non-healing ulcer. So we started with comparing the gene expression differentially expressed genes between each cell type individually, like fibroblast non-healing versus healing, NK cells non-healing versus healing, and so on. And from this analysis, we noticed that in the vascular endothelial cells, we can see there is vasculature development downregulation in the non-healing group. Like the genes responsible for vasculature development, we can see them here towards the downregulated side, not towards the upregulated side. And basically, those are the ordered differentially expressed genes from left to right. The top upregulated gene in the top left side, the top downregulated gene in the right side. So basically, the vasculature development genes are downregulated, which was expected from the data itself. This is healing group, this is non-healing group. But the next step is from the differentially expressed genes, there are secreted ligands and there are receptors, like transmembrane receptors. And from single cell data, we can identify the overexpressed ligand or receptor to try to infer the ligand receptor interaction between the different compartments. And for the healing versus non-healing group, we can see the different cell types can interact differently using ligand receptor interaction. And so subtracting the number of interaction between non-healing versus healing, we can see the differential number of interaction here. So here briefly, the red arrows indicate that there are more number of interaction in the non-healing side for those arrows. Blue means lower number of interactions in the non-healing group. But we can see here the macrophage, for example, have like all the incoming communication except one of them, for example, are more in the non-healing group. So like the macrophage has more incoming and more outgoing interaction in the non-healing. So we wanted to dig more in depth for the data to see what kind of interaction and what are the consequences of them. So we looked at the pathway level. Here we can see the green ones are exclusively happening, interaction exclusively happening in the healing group, not in the non-healing group, and the opposite in the below side here. And most of the interaction can happen in the both groups with different ratio. For example, the first one here, annexin. So this is a pair of ligand receptor interaction consisting of this ligand annexa1 gene and the receptor FBR1 gene. And we can see from the network that the ligand is expressed or secreted from almost all the cell types, and the receptor is expressed on the macrophage. And we found that this interaction only happening in the healing group, not in the non-healing group. The ligand itself is expressed in both groups, no difference. But for the receptor, it is expressed in the macrophage of the healing group more than the macrophage of the non-healing group. And from the literature, we can see that this FBR1, this gene or this receptor promote wound healing in different tissues, like in skin, in lung, in intestine, in different tissues in the body. On the other hand, there is another interaction that is exclusively happening in the non-healing group or more relative to the healing group, including this ligand receptor, TLR4 receptor on the macrophage, because it is more expressed in the non-healing group. And this interaction we found in the literature that TLR4 upregulation can cause diabetic peripheral neuropathy and retinopathy as well. And its knockout in diabetic ischemic wounds in mice can improve the healing rates. Finally, we not only the receptor on top of macrophage we can see, but also the secreted molecules from macrophage. We can see here this interaction, EGF signaling network secreted from macrophage towards fibroblast, keratinocyte, vascular endothelial cell, mainly the epithelial cell compartment. And those are the ligand receptor interaction within this signaling network. We can see ER, EG, those are all growth factors. And we can see their expression level mainly in the diabetic skin with healing diabetic foot ulcer, not even in healthy or diabetic without ulcer or in the non-healing group. And again, from the literature, we can see that this growth factor, for example, induces epithelial cell proliferation and differentiation, and the other one accelerates the keratinocyte migration and skin wound healing. The point from this analysis is not to identify a specific target or specific receptor or a specific molecule to upregulate or to downregulate, because here at the level of macrophage alone, there are a lot of more incoming communication and outgoing interactions from macrophage to other cell sites, and many of them are differentially expressed between the two groups. What about the other cell types? Like we have mainly 8 main cell types in this data or more, so we have a lot of alterations happening in the microenvironment of the ulcer. So it is not about specific molecule we need to target. This is why the next aim of this study was to identify a global, what are master regulators that can to try to change the gene regulatory network from a state to another state, from the non-healing state towards the healing state. So I will tell you very briefly about the approach. So we wanted to infer a gene regulatory network. 2 minutes. Okay. So I will skip the details of how to get the gene regulatory network, the approach, but briefly, we at each cell type level, we identify a gene regulatory network from the data, and the module in this kind of data, it is basically a transcription factor along with its target genes. And from those modules, we are hypothesizing that the downregulated modules, which is a transcription factor along its with its target, in the non-healing fibroblast, for example, need to be upregulated in order for the fibroblast in the non-healing group to behave like the fibroblast in the healing group. So we did the same for 6 cell types differently, and we identified the downregulated modules in all the cell types simultaneously, trying to upregulate them. And basically, for each transcription factor, we can identify a score for it based on the expression level of the target genes of it. And for those target genes, if there are transcription factor in them, and those also have targets, so we can identify also the expression, the score for each of them based on the expression level of the target genes for them. And finally, we made an equation to calculate adjusted score for the master regulator here, which is this transcription factor, to account for the direct and the indirect target. And briefly, this is the result so far we got. So this is one cell type, and the first column in the left is the score for this transcription factor depending on the direct targets only, the middle column depending on the indirect targets, and the last column depending on the average between them. So here, along all the cell types, the top 4 transcription factors we have, for example, those 4, have not only they are downregulated, but their targets are also downregulated, and the targets of their targets are also downregulated. So it is a large cascade of downregulation, and the significance of them, we can see that they can target genes involving in vasculature development, and the interaction which I mentioned a few minutes ago about FPR1 that can induce wound healing, it is also targeted or potentially targeted from one of them. On the other hand, the upregulated transcription factors, including the glucocorticoid receptor, which is known to have effect or to delay the wound healing in this data. And this is the summary of the study, that we identified several alterations in multiple cell types, and instead of targeting specific interaction or specific module, so we are trying to infer a gene regulatory network change across all the cell types simultaneously, aiming to perform cell reprogramming from the non-healing states towards the healing states. And hopefully or fortunately, we will see wound healing can happen in the non-healing ulcer. And thank you so much. Thank you.
Good afternoon. Thank you for presenting me, and I'd like to thank the organizers for inviting me for the second time to this event. I am going to talk about genetic variation. I am going to talk about genetic variation among Egyptians. Since the involvement of genetic factors in the pathogenesis of several diseases become important to study. 90% of genetic variations are in the form of single nucleotide polymorphisms, which can be divided into coding regions or non-coding regions, SNPs. Many important databases are available that collect many information about several genetic variants, including ensemble database of single nucleotide polymorphism, and cosmic database. One of the importance of studying the genetic variation among individuals is to link with the disease risk. As we can see, there are different expressions like benign variant, likely benign variant of uncertain significance, likely pathogenic, and pathogenic variant, through their impact on gene expression, protein expression, and finally risk for disease. This slide بتلخص importance of SNP genotyping. Number one, haplotype mapping linkages equilibrium analysis. Number two, population genetic analysis, phylogenetic reconstruction. Number three, disease management to find if there is a link or a correlation between the studied SNP and the severity of disease, besides screening for drug resistance among these patients carrying these types of SNPs. All of this is important for the personalized medicine. First, to predict disease susceptibility among these patients with SNPs, and to find drug-gene interaction and diet interaction through pharmacogenomics, pharmacogenetics, and nutrigenetics studies. The famous example is patients with lactose intolerance with single nucleotide polymorphism in lactase gene. Regarding calculating the appropriate sample size and performing the statistical analysis to report our studies or our results, sample size can be calculated using several softwares. One of the important or famous one is G-Power software, where we can calculate the appropriate sample size based on the power of study to be more than 80%, type 1 error, type 2 error, the type of study that we will focus, and choosing the appropriate statistical analysis to end with the appropriate sample size to document our results. After performing genotyping analysis, either by PCR reflex technique or real-time PCR, we have to calculate genotype frequency, allele frequency, and applying them in Hardy-Weinberg equation to filter out variants with possible genotype calling errors. One of the important statistical analysis that should be performed is binary logistic regression analysis to study the association of the studied SNP at different genetic models, homozygous, heterozygous, dominant, and recessive models, and the susceptibility to the disease to conclude if the SNP is a risk factor for the disease or not. The data are represented in the form of odds ratio, which must be adjusted by the known risk factors for the disease, such as age, gender, family history. All of this depends on the type of disease, of course. Finally, to adjust the p-value, we must make Bonferroni correction. Another statistical analysis should be performed is multiple linear regression analysis to find if there is a correlation between the studied single nucleotide polymorphisms or genetic variation and the clinopathological manifestations of disease. Now I'm going to summarize part of our published articles. Recently, in 2021, we published an article in Journal of Genetic Engineering and Biotechnology. The article titled Association Study of Hypoxia-Inducible Factor 1 Alpha RS115-49465 and Vascular Endothelial Growth Factor RS3025039 Genetic Variants with Diabetic Retinopathy in Egyptian Patients, cross-links with angiogenic, inflammatory, and anti-inflammatory markers. This work was in a collaboration with research of Ophthalmology Institute in Egypt. We choose these two SNPs based on the literature because the gene or the protein of HIF-1 alpha is important for oxygen homeostasis in the eye of diabetic patients, and vascular endothelial growth factor is important in the angiogenesis process. We had 4 groups: control, the type 2 diabetic retinopathy without type 2 diabetic patients without retinopathy, type 2 diabetic patients with non-proliferative retinopathy, and finally type 2 diabetic patients with proliferative retinopathy. Each group consisted of 72 participants. Genotyping was performed using PCR reflex technique. This table showed the distribution of the genotypes of the studied SNPs. As we can see in the highlighted part, the p-value, the genotype distribution of SNP in HIF-1 alpha was significantly different between the studied groups, non-PGR and PGR, compared to both type 2 diabetic patients without retinopathy and the control group. Also, the allele frequency was significantly different. On the other hand, the genotype distribution of the vascular endothelial growth factor studied SNP showed non-significant between the studied groups, except for the allele frequency, which was significantly different in PGR group compared to the control group. By making the association between the studied SNPs and different genetic models and the susceptibility to the diabetic retinopathy among type 2 diabetic patients, by performing binary logistic regression analysis, we found only significant association between the recessive model TT of the studied SNP in HIF-1 alpha gene at p-value less than 0.025 after Benfroni correction. As we can see, the odds ratio was adjusted for the known risk factors for retinopathy, which are gender, age, body mass index, lipid profile, and diabetic profile. In addition, we determined the concentration or the levels of some inflammatory and anti-inflammatory markers in the serum of the studied participants. As we can see, all groups, all diabetic groups with or without retinopathy, having increased inflammatory markers and decreased anti-inflammatory marker in the form of CTRB3 level. We made multiple linear regression analysis to study the association or the correlation of the side SNP with their protein levels, and we found a significant correlation between the side SNP and the protein level of HIF-1 alpha in the serum of patients at a p-value less than 0.0001, but no significant correlation was found between the studied SNP in vascular endothelial growth factor and its protein level. This figure illustrating the relationship between the serum levels of the studied angiogenic inflammatory and anti-inflammatory markers with the genotypes of studied SNP in HIF-1 alpha gene. As we can see in the red colors, there are differences between the studied groups and genotype carriers. But in vascular endothelial growth factors, there was no statistical significance in the studied parameters and the genotypes, except for PGR patients carrying TT genotype for CTRB3 level. Conclusion: we concluded that genetic variant in HIF-1 alpha RS11549465 could be considered a risk factor for non-PGR or non-proliferative diabetic retinopathy susceptibility among type 2 diabetic patients. This association might protect these patients or non-PGR patients from progression to the severe stage proliferative 1, even after long-lasting diabetes, 16 years, because of the increasing level of HIF-1 alpha protein with serine instead of proline at position number 582. This protein is known to be resistant to hyperglycemia and hyperlipidemia-induced disactivity in the protein function. Although this protective effect, it couldn't counteract the initiation of non-PGR among type 2 diabetic patients because of the severe elevations in the inflammatory markers and severe reduction in the anti-inflammatory CTRB3 level. On the other hand, this variant couldn't be considered a risk factor for the progression of PDR among type 2 diabetic patients. Another research was recently published in 2022 in the same journal, Journal of Genetic Engineering and Biotechnology, titled Impact of RS110-24102 Bellica and RS3753841 collagen 11A1 signucleotide polymorphisms and serum level of oxidative stress markers on the risk of primary angle closure glaucoma. This was performed in collaboration with Research Institute of Ophthalmology, Egypt, and National Research Center. We had two groups, control and BFG group, case control study. We performed genotyping using real-time PCR, and we found no statistical difference in the genotype distribution of the two studied SNP among the two groups. And we found no association with the susceptibility of the disease among Egyptians, so we couldn't consider these SNPs as risk factors for BFG. What are the challenges that we met? When we decided to study a SNP in a single gene in a disease, we focus on the literature or the databases and human and projects of 1,000 genomes alpha project and HabMap, which lack any information about North African individuals' genetic makeup, especially Egypt. So there is a mandatory for involvement of Egyptian genome reference in the current genetic dataset to study the risk of these SNPs with our diseases in our community. Thank you.